Like on to share a screen so that we can begin in earnest. So she'll be talking about that within 1 hour and 1 hour, 15 minutes, and then we can have questions, and then we can move on to the next section. So over to you.
Okay, thank you very much, sir, for this opportunity and nice meeting you, Mr. Yusuf.
Nice
Okay,
meeting you.
so hi everyone. My name is Shikoni Alaita Comfort. You can call me Comfort or Alaita, whichever one of Shikoni. And I'll be taking us through the introduction to 60 minutes R&D microbiome analysis. So just give me some minute to share my screen so that we can
Um,
see.
I forgot to I forgot to do a proper introduction. Sorry, I'll cut in. So Miss Olaitan Shekoni is currently I have introduced you before to today's class. She was one of our interns, and she's a bright star. And okay. Don't let me go into personal description so that people will not hijack you from us. So she's presently a research scholar at the Center for Food Animal Health at Ohio State University, United States of America. So our research is focused on nanoparticle synthesis and nanoparticle and vaccine formulation for food treatment, for treatment of food animals. So what does that mean? Food animals like we are in entomology, entomophagy, and like so. She will do more discussion about that so during that presentation. So over to you, man. I've
Uh,
got in my cutting.
okay. Okay. He said it already, so there's nothing more for me to say about myself. So I'm just trying to share my screen so that. So,
Let me see from
can
here
you
if I
see
can.
my screen?
Yes, 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 yes. Yes, we
Okay.
can see your screen.
Okay. So hi everyone. My name is Comfort, and I'll be taking us through the introduction to um, microbiome. Well, I know that we're all kind of familiar with the Human Genome Project, which was in which different microbes were, they were able to like sequence the complete genome of human being. For this course, it's actually into three parts. The first part is when we'll be meeting our microbiomes, while the second part has to do with knowing whether these are they our friends or our enemies. While the third part is talking about other part of the human body when we can get what's it called our microbiomes. So excluding gastrointestinal. So the objective of this course is number one, to know the importance of microbiome and why we should study it, why 60 minutes. Also to be able to define the term microbiome in your own understanding. Once you can get what microbiome is all about, then you can break it down into your own words then. So another objective is to know more about recent hypotheses around the ways microbes are acquired and also how it may change due to lifestyle exposure. For instance, the gastrointestinal tract, GIT for short, microbiome has been described as an extremely plastic entity, and how this microbial profile may be similar or vary between individual microbes. Also, to know the different body size, different body. we have different part of the body, but each of these parts they have different, or should I say, unique microbiome. And even a single part of our body, like let me say, for instance, maybe your armpit or some private parts have a different microbial profile. And most importantly, another objective of this course is to appreciate the importance of studying microbiome. So it's not just about knowing, but appreciating what has microbiome done, especially when it comes to biomedical researches. It is really really important in biomedical researches. So here, this is the first part where I will be talking about uh, the human microbiome. That is, what is microbiome? Microbiome, like this is my own definition of what microbiome is. So you can just think of microbiome as a kind of community of organisms, of different microorganisms. It could be fungi, it could be bacteria, it could be viruses, right? So when you're saying community, a place in which you can find different things. Do you understand? So I said microbiome is the collective genetic material of the microbe in a specific environment. And most importantly, knowing the human, it could be animal or environmental microbiome is very vital in understanding the human genome, which means that maybe when they were doing the sequencing of the complete human genome, then they try to put so many factors into consideration in order to be successful in these projects. Do you understand? Until we have the available sequence of every part of the human. Also, the answer to why we need or want to study the human microbiome is all tied to the exciting word which is referred to as 16S. Hello, can you hear me? Hello?
Yes, we can hear you. Please go on.
Oh,
Thank you.
okay. Okay. So the answer to why, most of us will be thinking, okay, what is so special about these microbiomes and why do we have to study? Why do we need to study? Well, studying this microbiome, answering this question, the only way we can answer this question has to do with 16S. Because of 16S rRNA sequencing technology, it makes every question we have concerning how we get microbes, everything very easy to be answered. Also, we have a right here, it says 16 has an internal transcript spacer active ribosomal RNA sequencing are common applications sequencing method used to identify and to compare bacteria or fungi present in a given sample. So just imagine you're working in the lab and you were given a sample, like there's and you picked a sample from, let's say, from the farm. As in you work with poultry and you picked a sample from, one, you did necropsy for your chicken and you took some sample from different part of this, chicken. And you want to analyze the kind of microbes in it. So you don't want to go through the microbiology way, like you just want to go into the dry lab, and you don't know whether it is colonized by bacteria or fungi. So that is why we have 16 years. 16 years is majorly for bacterial sequencing. Why this other IT has sequencing is mainly for what is it called? Mainly for fungi, because our fungi are eukaryotic, while bacteria are prokaryotes. So right here it says the prokaryotic 16S rRNA gene is approximately 1,500 base pair long, with 9 variable regions interspersed between conserved regions. And variable regions of the 16S RNA gene are frequently used for phylogenetic classification of genus or species in divers. What I mean by this is by the time you're doing your sequencing and you sequence the organism and you're able to get the sequence, you want to do a kind of comparative study between maybe available data on your NCBI of what's it called, organisms that are closer to this particular organism you just sequence. So for you to be able to do that, we call it phylogenetics. So it shows you the hierarchy, like what is it called, the closeness of that particular organism you sequence to whatever available data we have online. A case study of that is when we're trying to compare the DNA of human and that of all these chimpanzee like Pantrodontids or macaca, nemestrina, all these gorillas. We realize that there's a kind of similarity. So in order to be able to understand, I think the last course we did, Dr. Ashraf tried to explain what phylogenetic analysis is all about. And I remember there he gave assignment there we should try to like find the phylogenetic analysis of some organism of interest. So, and here the ITS region of the rRNA system is commonly used for DNA marker for identifying fungal species in metagenomics sample. So right here, meeting our microbes. Now, conventionally, it has recently been reported that more than half of the human body is non-human. Interesting. So which is this? So let's assume this is a whole complete human being. Like this is you, this is me, and there is a recent study that's reported that more than half of our body is non-human. Then what is it like? That is the question. So according to Professor Rob Knight from the University of California, San Diego, during the interview, he said approximately 43% of our body is human. Sorry, why the other one? Okay, 43% of our body is human. Why the rest are microscopic colonies? That is interesting. Which is this? So if 43% of our body belongs to us as human, then the remaining population of our body belongs to microbial. We don't even know where they come from. We don't even know how they evolve. We don't even know anything about them, right? So right here, this is it. Where did this microbe come from? Well, it has been discovered that newborn babies are born completely germ-free, but the initial colonization of newborn occurred during the process of delivery. And right here, the process of delivery has been considered the primary contaminant of GTI protein profile. Several studies using 16S RNA gene sequence have shown that infant born vaginally acquires microbe through the mother vagina, while those born through C-sections acquires microbe through the mother's skin. And this would make sense. That means that when it comes to giving birth, we have to delivery mentor. You could give birth, or naturally, or you through the vagina, or layers, and there's some kind of complications, and you can give birth on your own. Then they can do a C-section for you. So now, if a child is born naturally, that is through the vagina, that means this baby would have the microbiome of this baby would be that from the mother's vagina, because that is where the baby is actually passing out through, right? Just the way we have this. Why they assume that the mother has to go through C-section before the baby can be delivered? That means that through the process of bringing out the baby will acquire the microbiome on the mother's skin. Example of that is the staphylococcus. We know that is the natural microbiota actually. For the moment the baby is being, delivered and bringing out and is in contact with the mother's skin and everything, the baby automatically get that. And this right here is just sequencing analysis that shows different what's it called? Different delivery. So right here, I was like, after a vaginal delivery, infant growth microbiome are typically colonized by maternal vaginal flora like Lactobacillus and Rhophtella species. In contrast, maternal skin flora are dominant after a cesarean delivery. Example is Clostridium, your Staphylococcus, your Propionibacterium, and Corynebacterium. So, which means that if the baby is being given birth to naturally through the vagina, the baby get this lactobacillus and this other organism as part of the microbiome. But if the baby has to go through C-section, that means the baby get all of these, all of these Clostridium, Staphylococcus, all of these organisms. So right here we have a different hypotheses that show this actually. So right here we have the mother body habitat. We have the oral mucosa, we have the vagina, and we have the skin. While we have the baby delivery mode, it could be vagina or it could be cesarean. So right here, there means the oral mucosa of the mother that's from, it could be from whatever the mother is eating to whatever the mother is drinking, everything, pregnant women during pregnancy, they take diet, it could be antibiotics, it could be placebo, whatever they're taking, like it goes through the oral cavity, right? And then down to the baby and whatever happens. For the point there is that during our delivery, we already know that this baby are actually born germ-free. But the only point in which they come in contact with microbiome has to do with this delivery mode, which could be, okay, at this microbiome, if this baby is being born vaginally, okay, what are the microbiome of the mother's vagina? Okay, if this baby is being born through operation, what are the microbiomes available on the mother's skin that this baby gets, right? So right here, this one actually explains it better. According to the gut microbiota of infant born vaginally, that is resemble the microbiota of the mother's vagina. That means let's assume, okay, this baby is being given birth to right now. Then I through vagina, then I took some sample from the baby. Then I went ahead and I took some sample from the mother vagina. And I want to do a kind of comparative study in order to know the type of microbes available in the mother's vagina and that of the newborn baby. So there's a study that says that the gut microbiota of an infant born vaginally resemble the microbiota of the mother's vagina. While the microbiota of infant born by cesarean sections are similar to the mother's skin microbiota. For yeah, another study has seen that recent study that uses 16S RNA sequencing have provided information on earlier microbial colonization in newborn, which means that previously we know that babies are born germ-free, and the only way they get microbiome has to do with the delivery mode, which could be vagina or through C-section. But now there is a recent study that is saying that most of the study that they used on 16S RNA sequencing to conduct has provided more information on microbial colonization in baby there, even before a baby is being given birth to. They're already colonized by microbes. And this kind of colonization is referred to in inter colonization according to this person, because this person carried out a research, right? So yeah, I said the intercolonization hypothesis proposed that some microbial members of the infant gut microbiome are acquired before birth, probably via contact with the placenta microbiome, which has been suggested to originate from the mother's gut or oral microbiome. So this diagram right here shows that this one shows us that this is a sterile womb. The fetus and the placenta are kind of sterile, and the gut microbiome is acquired after birth. We have this. Everything here looks, if you see this, sorry, I would like us to show some indication though. You with me. If you want me to go back, just kindly talk, okay? Am I talking to myself alone, please?
We are with
Please
you.
go ahead. Please go ahead. Go
Okay.
ahead.
Okay.
Thank you.
Thank you. So right here we can see that this baby, everything looks good. No germs, nothing. Right. Then the moment the baby is being given birth to through vagina, it goes to microbiomes, right? And the microbiome stays after delivery, stays with the baby, right? And here, a baby born through, through operation, this is when the baby get the what's it called? The baby get the microbiome from the mother's skin, and this stays with the baby. So right over here, in order to prove this in utero colonization hypothesis, you can see that right from the mother womb, the baby already has a microbiome. Can we all see that?
Yes.
Yeah.
Yeah. So the baby already has what's it called? These microbiomes and microbiomes right before the baby is even being given birth to. So it doesn't really matter this time around whether you're being given birth to vaginally or through C-section, because before you're being given birth to, naturally, right from the womb, you already got some, you're already colonized by some microbiomes, like the placenta. So here over here, I don't know if you can see that here over here is the placenta, and we can see that there's some colonization already there. So, and after the baby is being given birth to. So right here, so in support of the utero colonization hypothesis, bacteria has been identified in previously considered sterile sites, such as the amniotic fluid, the placenta, and the cord blood. That is, this is the placenta. This is the cord blood. So, and everything here, from here, right here, we can see the colonization. We can see that everything is already cloned even before the child is being given birth to. So, to better understand how in utero colonization may occur, numbers of hypotheses has been generated. One hypothesis says that the maternal vaginal microbiota ascend into the amniotic fluid, which is the first hypothesis. That means the maternal vaginal micro it goes into this amniotic fluid. Another hypothesis suggests that, well, maybe the maternal GIT microbiota uptakes via dendritic cells, which is this. And another one now says that after this maternal GIT microbiota uptake through the dendritic cell, then it's now being transferred through the placenta to the fetus. That is, okay, this one says that the whatever in the microbiota in the mother's vagina flows into the amniotic fluid. And you know the amniotic fluid is actually meant to be a sterile environment, like a sterile outside. Then another hypothesis came up to say that, okay, after this whole thing happened and explodes, then the maternal GIT microbiota uptakes through, that is also go through the dendritic cells. Then after it goes through the dendritic cell, it's yet to get to the baby. Then it's now being transferred through transplacenta to the maternal system that is after this stage it goes here, then after it goes here it goes here. You see that this actually goes into the placenta that connects the mother to the baby. So right there you realize that whatever the mother is carrying, everything now goes here. So all these sites, the site over here and the one over here that is actually meant to be sterile, that is your amniotic fluid, your placenta, or your cord blood, becomes colonized. So this hypothesis helps us to further understand that, oh, truly, before a baby it's not until a baby is kind of given birth to before he or she gets a microbiome. If right from the womb, the baby gets. So right here we have some studies. They study this a critical assessment of the sterile woman in utero colonization hypothesis implication for research on the perineal infant microbiome. So here we're 16 RNA sequencing technology has made a great contribution, which means that without 16 we wouldn't understand this. What we would know is that, oh, okay, when I give birth through vagina, my baby gets the microbiota from my vagina. So when I give birth through C-section, you get the microbiota from my skin. But we won't, without 16 egg, we won't be able to differentiate that. Okay, let me take a sample from this baby. Let me take it from the baby's gut. Let me take a sample from the mother. And let me make a comparative study to check if these organisms are kind of similar or are they different. Do we understand? We want to know. But without sustenance, we wouldn't have been able to do it. Without sustenance, all these hypotheses would not have been able to be proved. Well, so, but unlike any other techniques we have for sequencing and everything, the, the 16S RNA do not have the limitation in studying acquired microbiome from different parts of human. For instance, a study carried out by Lauder et al. 2016 compared the bacterial profile from vagina. That is, this man carried out an experiment. And what it did was it collected sample from 3 different parts. It collected the vaginal swab, which is a sample from the vagina, then it collected another sample from the saliva, then another sample from the placenta. The reason it was trying to do this was it was trying to check the microbiomes. These are different sites. Previously I said that one of the objectives of this course is to understand the the different part of the body where we can get different microbiomes, and in a particular part of the body you can have a distinguish a different and unique microbiome, right? Microbial community. So in order to be able to prove this, it took the sample from these 3 places. And after taking the sample, so this graph right here, this is the vagina. This right here, this over here is the vagina. Let me see. Is the vagina sample you took. Why this right here is the saliva sample you took. And this, the whole from here to here is the placenta. So after taking it's just like when you're carrying out your normal laboratory experiments, you always want to have a negative control. Let's say, for instance, I want to formulate a vaccine using nanoparticle. What I do, okay, I need to have a negative control, which could be a blank nanoparticle without any DNA or any RNA or any protein in it. Then, whenever maybe after formulating the vaccine, and I want to check the protein efficiency and everything, then maybe I want to use nano drop or PCA protein acid kit or whatever I want to do. I need a negative control. So my blank, my empty nanoparticle that has no protein, that has no DNA or RNA would be my negative control. The same way this muscle needed a negative control. So whatever you're doing, we need a negative control. So if I had a negative control, then it was trying to make a good observation that it realized that if you notice that right here, right over here, this sample, they kind of look similar. This is the negative control for his experiment. And this is the placenta sample. This is the saliva sample. You realize there is a little bit different between the saliva and also the vagina. But when it comes to the placenta and the negative control, look at it. They were kind of similar. Look, these all places are similar. This one right here, a little bit similarities, but still we can still give it about maybe 50%. And what's wrong? Okay, and this, then it realized that it found out that when comparing the bacterial profile to the sequence of the negative control, we found that the bacterial profile in the placenta resembled that of the negative control. And after this, then it brought just this is the placenta and the negative. So when you check it, you realize that the whole sample here, right here, looks all the same, which means that in this sequence, whatever microbiomes in the blood solution used as its negative control are the same bacteria in the placenta of a sample. Then, so this study help us to understand that maybe okay, this 16S RNA gene sequencing study design should be conducted with care. It could be maybe the reason he had is or it could be maybe contamination when he was trying to make his study design, you know, the kind of what's called sequencing and everything he wants to use, the reagents, everything he needed. Maybe there was a kind of contaminant. It could be a mistake. It could be anything, right? So this is not suggesting that this studies is suggesting that the 16S RNA gene sequence is not something you can just do in a rush. Oh, I just want to do it for doing sake so that I can tell everyone that, oh, I can do 16S RNA sequence. No, because if we do it anyhow, you will never get the result. Like the result will be kind of biased. So we want to prevent that from happening. So which means that it should be handled and conducted with care. And the sequencing control should be included. That means if there's a particular what's it called reagent you're meant to use, then I shouldn't try to improvise and do something because I will never get the same answer. So right here, however, despite the call for concern when using the 16S RNA gene sequencing techniques to study low biomass sample, example of a low biomass sample includes placenta, the amniotic fluid, the cord blood. They have low biomass samples. They have so pregnant Ductus placenta, pregnant mother received probiotic compared to the placebo control. Bacterial alteration was discovered in the placenta as well as the infant meconium. Okay, right here, we can see that this woman, should I say this case? Okay, this woman actually took some probiotics during the pregnancy, and the baby got the probiotic. Why this over here, this person took a placebo. Placebo is just like a pills, like used for it's being described for maybe some for some physio or whatever analysis. Maybe your doctor prescribed it for you that you should use it, or you yourself will what you be doing pregnancy. It doesn't have anything to you, but some people use sometimes some people use it to prevent pregnancy, like overdose of it, something like that. So you can see that this woman took the probiotic here and this woman here took the placebo here. So this placebo is just like a control, while this is, we could call this a positive control. So we're trying to like compare the two of them. So what we realize is that there was actually a bacterial alteration in the woman that used the probiotics, as well as in the infant and microbial over here. So here, this finding actually to support the notion of the in uterus colonization study carried out by Jimenez et al. 2005, where this person took a genetic label enterococcus strain and which could be isolated from the microbiome sample. So this mice, or should I call this mouse we have this mummy right here. This mummy is actually what's it called? As a is a labeled as a labeled enterococcus strain in it. And we realize that what's it called? So we said that the microbial sample from the offspring of the mice of the orally inoculated pregnancy mouse. So this mom, she's pregnant and being injected with what's it called, enterococcus strain. But by the time she delivered, all her offspring, they have the same. Look at it right here. They have the same microbiome. So it was actually two groups. One is the labeled group, while the other one is the unlabeled group, the uninoculated group. So this group is the control group. This group over here is the control group, while this is the real experimental group. So when this mommy gave birth to her offspring, they had the enterococcus. But this particular nigger right here, look at it. The nigger wasn't inoculated with anything. So by the time she gave birth, they had nothing. They couldn't find the trace of this enterococcus strain in this one because this was actually the nigger. So all of these hypotheses is just trying to prove the fact that we're trying to prove the in interest hypothesis, colonization hypothesis that what is it called? Before a baby is being given birth to, there is a colonization right in the womb, and this could be due to the microbiota from the mother's guts. Do we understand? So, in summary of this first part, we are more microbes than human, and also studies using sustained sequence technology have shown that we may acquire our microbe during the process of delivery, and that profile may be dependent on delivery mode. In addition, studies which include study using 16S technology have recently reported that in utero colonization may occur prior to colonization during the process of delivery, which means that even before a child is being given birth to, the child has some colonization and microbiome colonization even after the baby is being given birth to. So it is now left to now compare these two, whether I'm being given birth to vaginally. Let me check. I'll take some sample and check that. Is it similar? Then I'll take some sample from the placenta itself to make some comparison and see that okay, does this baby still have those microbiome, like the microbiotas in the placenta? And lastly, studies investigating sample size such as placenta sample, cord blood, and amniotic fluid, which are all regarded as low biomass specimen, needs to have optimal study design and sequencing control in place because we can just sequence and analyze for doing sake without making sure there maybe good laboratory etiquette and kind of put in place. Like everything we need should be in place. Now there, oh, I need to go find a primer. Oh, I need to go look for these. Oh, I need to take the micropaper. Like everything we need to carry out a successful study should be in place because these specimen, or should I say this sample, are actually low biomass, and every little thing in them will give you a false result. So that is the end of our first part of meeting our microbiome. So the second part here is knowing whether these microbiomes are they our friends or enemy. We want to know, right? So right here we have this mommy. It's a pregnant mommy. So in this file, we will further discuss about number one, how early life of gut microbiomes might change over time. And also we'll try to look into how changes due to different exposure in life may impact on health. So, and like I said, in part 1.1, we discussed about the mode of delivery as in early life exposure, which may modulate or influence the gut microbiome since we might be acquiring our microbes in utero. And also, a number of prenatal exposure has been shown to modulate this microbial profile. For instance, gestational duration, prenatal antibiotic treatments, maternal prenatal stress, maternal diabetes status. All of these are prenatal exposure, which has shown to actually modulate these, our microbiota profile. So also our daily life exposure, such as what we eat. That was why I was trying to say previously that maybe what the mother takes, the diet, the food, whatever she drinks, actually have effects on the baby. So this could be one of the reasons why the uterus is kind of colonized even before the baby is given birth to. So, so we have a mode of feeding. It could be milk feeding, breastfeeding, antibiotics used, solid food, antibiotics, all of these actually have an effect in what's it called, in modulating our microbiome profile. So right here, so this is our microbiome is like an eating organ. So what it does is that it helps us to break down food stuff that our own cell cannot cope with. So just imagine that you give birth to a baby and the baby is like 2 weeks old. And you want to give the baby rice to eat. Is it possible? Hello, Terry, can
No,
we
no,
find
no,
No, it's not possible.
no,
It's not
no,
possible.
it's not
It's not
possible.
possible.
it's a little bit, we have to put our own talking to myself again. Anyone, so it is not, we know it is not possible, right? So what our microbiome does is that what we can do, what our normal cells can do, they help us do it. That is when we feed them with the right thing. So, and in turn, our food affects our microbiome. So, and our first set is laden with gene for digesting milk protein. And that is when this baby keeps sucking from the mother. It's not that the baby can instantly digest the milk like that. It is the microbiome that is present in the baby that helps in aiding the digestion, like breaking it down to what the baby needs. So allowing us to make full use of our only source of nourishment as baby. And as baby, maybe 2 weeks, 3 months, 9 months. I think there are some people that even till 3 years they're still sucking the mothers, they're still breastfeeding them, right? So all that is our nourishment. That is the food we rely on. That is what we depend on for at that stage, let's say maybe 3 months or 6 months, we can our cell is not well grown enough to help us to break it down. So it is the available microbiome you gather because we try to see the advantage part of this is that they help us to break it down and make it go to different part of the body that it is needed to give us energy to kick, to cry, to do anything we want to do. Also, just before we move into solid food, which is this, our microbiome start activating gene that breaks down the complex sugar and starches in plants, preparing us for the menu to come. As our diets kind of diversify, so do our bacteria. So as whatever you're what is it called, eating, what you're taking in, as it starts changing, it's actually affect the bacteria, like your microbiome, the bacteria. So they keep evolving, they keep growing, they keep changing. So you can compare uh, the microbiome you had when you were kind of 6 months with that of the one you have when you're 1 years old, because you've taken a lot of nutritious food, right? So you expect them to also grow because the way they grow, as they grow, they help you to fight against some germs. They help you to build your immune system, right? Because in your immune, they help you to fight off invaders in the immune, they help you to activate your T cells. So your T cells send signal to your memory B cell. That's what they used to say that if a baby have smallpox, and the baby got rid of it, the baby can never and ever have that smallpox again. The reason is that the moment this microbes is coming in to invade your immune system, your T cell, what it does is that send message to your B cells, which is your memory B cells, that hey, there is someone trying to create news and say, I need you. So the B cell, what the B cell does is that start checking a record or his record. Oh, do I know this nigger? Have I seen this nigger before? So, oh yeah, I've seen this person. So they start helping you to secure what antibodies to fight against that invader. And before you know it, it goes. But in a way, in such a way that you are now feeding yourself, like you're not eating proper food or anything. Let's just use the case of all these malnourished children. You see that they are actually prone to various diseases, like various diseases, because the microbiomes in them that is actually meant to act as a defense mechanism for them is weak. So they're like maybe 6 years old, but the microbiome is still like 2 weeks old because they are not feeding well. So how do we expect the organism in them to grow well, to strive well? So also they activate genes that use carbohydrate effectively, produce vitamins, and break down unusual and diverse chemicals. As adults, our microbiome becomes relatively stable, but its membership roster depends on the food we eat. So, as in you're not feeding well, you're not taking the necessary things you ought to take. How do you want this to be stable as an adult? It's not possible. So right here, as we learn more about our bacterial partner, we might eventually find ways of influencing them to improve our health. This is already happening. In 2008, Alexander COHORT from the University of Minnesota managed to cure a woman with a vicious gut infection by giving her a transplant of her husband's gut bacteria, which means that they are not, we don't know if they are enemy here. We know that they've been doing something nice. They help us to carry out some metabolic activities in our body. They help us to break down some chemicals, some food complex that our cells can do. They perform so many tasks that our normal cells cannot do. But so this particular study carried out by Alexander actually proved that they are our friend. So if this woman is having a serious and chronic gut infection, can you imagine that after taking a transplant of her husband gut bacteria, she got treated and she got rid of the infection? That shows how important and how beneficial they can be. And that was what I said the other time. They have diverse of function. But right here, so the microbe is more than just our partner in digestion. They affect our health too. And the moment they start affecting, if you do not eat properly, your immune system will be weak. And this will have a great effect on our health because you feel weak, even if you'll be prone and exposed, you'll be vulnerable to different infection. You'll be prone to different invasion of pathogenic microbes, and it causes serious damage. So they have been linked to a variety of medical conditions, including allergies, immune diseases, and even obesity. For example, the balance of the two major groups, the bacteroidetes and the firmicutes, could influence our body weight. So these two organisms they actually influence our what's it called obesity, because it is now until because obesity is also part of malnutrition. It is now healthy, and if you notice, those are people obese. They have various health challenges, diabetics that are prone to it, rheumatism that are prone to it. So right here, the bacteria of our microbiome are mostly our allies, but they can also be turned against us. The 2-year study in mice have found that viruses, including the one that causes polio and another that causes cancer, can exploit gut bacteria to infect our body. So you can see this little finger right here. Okay, she has cold, she's sneezing, she has asthma. This niggle right here having stomach ache. It could be ulcer, it could be anything. And this little cute baby right here is suffering from some serious immunological diseases, right? So here, C-section of vaginal delivery, for example, has been associated with an increased risk of immune disorder such as allergic reaction, asthma, and other immunological diseases. And what the statement means is that children that are kind of given birth vaginally kind of have a better microbiome that helps them to fight against germs compared to those that are born through C-section. And that is why we have a new study that is doing, we call it vagina seeding. What it means is that they take out some, what is it called, the microbiomes present in the mother vagina, and immediately the child is being given birth, so maybe through C-section. So they go to the mother's vagina, they take the, what's it called, the microbiome readily available there, and they seed it from the baby. The reason they're trying to do this is that they're trying to make sure that when this baby grows, there will be a kind of decrease in, should I say, health challenge. Because scientifically, children that are being given birth to vaginally, they kind of have better microbial population that helps them to fight against other pathogenic microbes that can cause serious health issues compared to those that are not born through the mode of delivery. So, in summary, the GIT microbiome is a plastic entity which is modulated by a number of exposure throughout our lives. Large numbers of sustained studies have contributed to our current knowledge of the GIT microbiome, which has led to a number of potential interventions for disease states. To date, the majority of sustained studies are focused on the GIT microbiome. However, this research is still very new and more well-designed studies are needed to better understand not only what is there, but also what they are doing. So this is the end of part two for this course. So before I move to the last part, any questions so far? Can I proceed?
Yes, please.
And I can hear just one person.
Hello, sorry.
There are questions
Yes.
Okay.
from Mrs. Dr. Merit.
Okay, good morning. Thank you so much for the presentation. Um, I would just like to start by saying that from there was a quote you made by one professor from one university. Sorry, I can't remember the particular
Oh,
university. Let me
I,
just quote it. You
okay.
said, "Can you hear me, ma?"
Yes, yes, ma'am. I can.
All right. So you said approximately. I mean, the professor said that approximately 43% of the body is human, while the rest are non-human, that they are microbes or
Yeah.
microbiomes and that's where. So I wanted to just say that how do I link this up with the questions I want to now ask you? What's the implication of acquiring this knowledge on microbiome? I mean, what's the application of this microbiome in biomedical research? Other examples apart from this fetal environment you mentioned. I mean, are there different pathological conditions that I can link it up with? And then, what do you mean by colonization of the microbiome in a fetal environment? And also, what's the difference between colonization and then the in utero colonization during the process of delivery? So that's these are the questions from the first part. I hope you'll be able to remember because I want to, from this second presentation you gave, I have other questions, but I think let me hold on and allow you to answer the first set of questions before I go ahead to ask the other questions. Thank you.
Okay. Thank you, ma. So this is what you're gonna do for me. the question you ask now, you ask them one by one. So once I give you the answer, so you ask another one. Can you do that for me, please?
Yes, please.
Okay,
All
so
right. So
once you
I
go
said,
to
yeah. So the first one, what's the implication of this? What's the implication of acquiring this knowledge? How can you help me to um link it up?
The first was the quote by the professor,
Yeah,
Professor
yes, yeah, I
Robert.
said that quote, how will it help me to
43%
understand this question? Yeah,
human.
yes, this one
I think
exactly.
it's Professor Robert from University of UC Berkeley.
yeah.
Uh,
Yeah, that's the
No,
black
this is
hole.
this
That's
particular
it. Yeah,
slide, this one, this is it exactly.
yeah, this all right.
Approximately, yes, this one particularly,
Okay,
yes. Yeah.
okay, thank you. So if
Next question.
this professor is saying, sorry, please let me answer this one so that I wouldn't forget. If this professor is saying that 43% of the human body is human, why the other part of the body is microbial colonies, which means that you realize that normally some people they don't have eczema, and medically, all these facial treatments, all these facial cleanser, it's not really good to use them because the moment you're using them, you're killing the microorganism there. And what do? They help you to fight off any other microbes that can cause maybe skin rashes, maybe all these dermatological diseases on your skin. So they are there. Okay, for instance, thank God you're a lady. In the vagina, we have the normal microbes that what do? They prevent you from having vaginal infection. And that is why the doctors will tell you that you shouldn't use soap and all these detergent or solvent to wash your private part. What they're trying to do is that the moment you're using those soap to wash them, you're killing your soldiers, you're killing your armies that are there to say guide you. So the knowledge of this is that for every part of your body, you have some microorganisms that maybe that particular part of the body there is the natural, what's it called, natural habitats. They stay there and they keep up any other pathogen that might want to call come there to cause maybe anything in you. For instance, when I was talking the other time, I tried to talk about the fact that even inside in your what's it called, when you're eating in your lung, you have some microbes that is the natural habitat. They aid the secretion of your saliva, they aid the activation of some hormones that helps in the secretion of your saliva, which you as a person need to digest your food. So this knowledge you can use it in diverse of ways. But the first thing is related to yourself. Do you understand? The only way the staphylococcus on your skin becomes an infection is when it gets into an environment that is not the natural habitat. That is, let's assume you have a cut on your skin. The moment you have a cut, it's leaving that is natural environment into a new world that everything it needs to strive for is there. And before you know it, because that place is not where they're meant to be, they start causing serious infections. Like, are we good?
Yes, thank you very much.
Okay, second.
Um, yeah, the second is okay, let me just say I think you've answered the implication of this. You've answered that as in personal application. I need to apply it to myself. So I wanted to ask, so what's the application of this in biomedical research apart from the fetal environment example illustration that you gave? How can it be applied in the light of, I mean, other pathological condition of interests,
Okay,
Yeah.
good. So let's assume let me use cancer research as a case study, or don't let me go too far. Okay, let me use let's assume your okay. Let me use agricultural sector. We know that there is this bacterial Bacillus thuringiensis, right? It's a very good microbe that has a good they have these pesticide agents in them. They are used as biopesticide, right? So You isolated, and the natural habitat is the soil. But one good thing about them is that Bacillus thuringiensis and Bacillus cereus are kind of very similar morphological. So you want to differentiate between both of them, but you don't know how to. But you know that they have what's it called, like some kind of similar characteristics. But one good thing about Bacillus thuringiensis is that they have cryo proteins, and this cryo proteins is the one responsible for the biopesticidal activity. How do you sequence it out? You can only sequence it through the help of 16S RNA sequencing technology. Like, am I communicating? So when it comes to maybe let's say you're working on bacteria and you want to do the metagenomic analysis of this, you use 16S RNA. That is a very good sequencing techniques that help you to do whatever sequencing you want to do on your bacteria. So there are ways in which this knowledge can be applicable when it comes to maybe environmental bioremediation. We have some bacteria that are good bioremediation agent, but you want to sequence them. You want to make sure that you have let's say someone found out that this particular Bacillus thuringiensis with this, let's assume this strain K2 is a good pesticider. We need a novel strain of this same organism. So I took a sample from Saudi Arabia and I got a K2 strain, and I took another sample from Abuja, Nigeria, and I want to know the strain of this new organism. Is it the same or something that nobody has found out? Maybe a particular strain that no one has found out before. How do I do this? I use 16S. And by the time I use 16S to sequence out the gene, the next thing I want to do is that I want to do the final genetic analysis to compare, to make comparison. Have I found something novel that no one has ever done before? Are we communicating? So there
Yes,
are diverse of ways we can use this, especially when you're dealing with bacterial specimen. The best techniques to use is 16S. And that is why it is important to have a basic understanding of what this techniques is all about. What is 16S? Why do I need it? If you can answer this question, and let's say you're done with the wet lab analysis, the microbial, the way the world is advancing now, you don't need to start culturing your organism and everything before you know. Especially when you know that you're dealing with bacteria, what is it called, specimen. You don't need to go through the wet lab. That you have to culture, do serial dilation, do all of this. There are new techniques, advanced techniques that helps you to sequence them out immediately, and it's time effective and efficient. And one of them is 16S, especially for bacteria. So, have I answered your
Can
question?
I come in
Yes, sir,
so that
please.
to also um, shed more light and also give more direction. For those who have questions, please, if it is possible for you to write it out so that we can read it once and for all. And to add to the answer, 16S application, 16S microbial RNA, its application is very vast. So, and she, resource person, has given us an example of that. So, we use it in agriculture, just like the BT, and we also use it in medical entomology also for pest control and prevention of the transmission of disease. For example, I made mention of the fact that I'm also working on 16S RNA. And what we do majorly is that we look for the microbiome that affects insect, and one of the that affects mosquito in particular. One of the most popular one is the Wolbachia. So Wolbachia is one of the most effective microbiome, and we've noticed that we can manipulate the life of the insect by manipulating the microbial community it has. We've noticed that the microbial community plays a lot of role in vector transmission, vector capacity, disease resistance, insecticidal resistance, feeding, mating, choosing evolutionary. So basically, what as it is happening in human being, and just like it was explained to us earlier also, our body is made up of majorly microbiome. So microbiome is the second, is even the largest DNA component of our body. Like if they sequence the microbiome community of everyone, so we have everybody has unique fingerprints of microbiome component. So the application is so wide in human. We have different lab. Dr. Abdul Qadir will be the next presenter. He's also working on 16S microbiome, and he chose a particular section. I am working on the insect part. Miss Olaiton is working on another part. Dr. Abdulkadir is also working on another part. And we have different researchers working on different parts. There's a laboratory in my university that we have those that are working on the food microbiome, soil microbiome, gut microbiome, environmental microbiome, plant microbiome. A lot of, and now I'm working on insect microbiome. So it is so vast and so magnanimous that you have a lot of application in that. So because of the role they play in the ecology, evolution, and human or insect or living organisms life, so they have application. Yes, there is a connection between the microbiome body and the immune system. Just like it was explained to us, what's one of the roles that the microbiome does is that it protects the body from harm. So if we are, and another aspect that is very juicy for those of us who want to work on that is the aspect of microbiome, 16S microbiome, and antimicrobial resistance. So we noticed that, okay, this is the work of Ms. Olaito. So, but notwithstanding, let me quickly write it up so that the you notice that when people use antibacterial drug, antibacterial, so you wipe out that, and that's what she has been explaining from since morning actually. So you wipe out all the communities of the both the beneficial and the not beneficial. And there is a condition we call dysbiosis. The dysbiosis is the state of imbalance between of microbiome in your body. When there is an dysbiosis in your body, you will fall sick. And as a matter of immune resistance, when there is a dysbiosis in the body regarding microbiome composition, there is a disease occurrence, especially those that are autoimmune diseases, obesity, diabetes. So a lot of things that I would implore you that you should explore that field of microbiome analysis, 16S microbiome analysis. So it's a field that you will always be swimming, and you might not swim and complete the swimming through. So I want to take one or two questions, and I want resource person to also answer that so that we can pity how would I put it, so that we can move on to the next one, because we still have a lot of things to discuss, and we don't want to keep everyone waiting and delaying our time. So any more questions?
I think someone asked a question in the chat.
Chat box?
Yeah, there are we promoting? No, we're not promoting C-section. Please don't get me wrong. I was only trying to explain the two type of delivery model. We have the C-section and we have the vagina. But in fact, the vagina is more better than the C-section because of the kind of microbiomes you get. Like, am I clear on that? Please
Yes.
don't get me wrong. We're not
And it
promoting.
has been studied that those who are given birth through by C-sections, they are prone to
Yeah, well,
immune-related diseases in the
disease.
future.
Yeah, they have.
Yeah. So
And that is why they're doing this vaginal seeding now, where they take the microbiomes from the woman, from the mother's vagina, and seed it on the baby to prevent this, especially that is the baby that is being given birth to C-section.
C-section. Yeah.
Okay, so is there a link or connection between microbiome study and the immune system? Yeah, yes, ma'am. Yes, there is a link. There is a link, and I think I already answered that. They had like your soldier, the way we have all this army fighting for Nigeria, right? That is how they are for you in your body. So there is a role because they help to activate your immunoglobulin A that is found in your dendritic cells. That is more of immunology. So let's please not go deep into that. So there is. So is there a plan to use the microbiome research in the treatment of
Sorry,
infectious
sorry. Let me
disease?
take you back a little bit.
Oh, okay.
Sorry.
Okay.
I just wanted to hear what I didn't get the last statement. Dr. Ashraf said. He said those giving birth to C-section has what something I didn't
Okay.
get it. I didn't get.
Okay. So
So
I
they
want.
have there, there
Okay.
there is a potential that they will suffer. They are more prone to infectious diseases than those that are given birth to by natural vaginal birth, and it has been
Okay.
proven severally. So that's why those who give birth to C-sections, they do also advise them to do more of breastfeeding to improve the immunity status. But they can never have the same microbial community that because it's like getting to a barren land. The first person
Mm,
that colonized the place would always be the owner till eternity.
Mhm.
So that is what happens with a bacterial community from the vaginal and also from the skin. For those that are from the C-sections, they are the microbial community that colonize them most are those from the skin, while
yeah.
that from normal birth is from the vagina. So there is difference between the two, as
Thank
explained
you.
by facilitator.
Thank you so much.
Please,
Okay, sir.
before we move on, please.
We have two more questions. After that, two more questions, we move on to
No,
the
no, no,
next
no. It's not
presentation,
a
please.
question on a particular term, please. We're talking
Mm.
about in like I don't know. Is it that after the baby has been born, they will kind of spray the microbiome in his body, or what's the meaning of?
No, no, don't get it wrong. This is what I'm trying to say. There is this new research that there it is called vaginal seeding, and what it means is that um before
EDIM.
before the baby is being delivered through C-section, they already take the doctors and how they do they wear glove and insert it into the mother's vagina and leave it there for at least 30 minutes. So after 30 minutes, they bring it out and put it in a sterile container. So the moment the baby is given birth to through C-section, they coat the baby with this sterile glove that is full of the mother's microbiome, like amicalae. So this you can read on it. It is called vaginal seeding. It's a new what's it called? It's a new area of research that people kind of interested in, especially when it comes to microbiome and success analysis, stuff like that.
Sorry, you said the
So
name is vaginal seeding.
yeah, vaginal seeding.
Seeding. Okay.
Yeah, it is.
One last question.
Okay, is there a plan to use the microbiome research in the treatment of infectious disease? Yes, that is the gospel. We've been gospel of science we've been preaching since morning. Yeah, it is very very relevant when it comes to treatment and control of infectious disease because it makes you understand better the cause of the disease, the organism involved, and it makes you to make a comparative study between any other thing you want to do. So it's really,
Especially malaria.
yeah. So the malaria man is talking.
Yes, so
Okay, so they have probiotic the yeast base does 16S. Well, for this one, they use the other ITS rRNA sequencing. In the first part of this course, I talked about two type of sequences. 16S. I said 16S is mainly for prokaryotes. Why? What's it called? The ITS is made for the eukaryotic, so that your fungal, your yeast, and all things. So it's used. I hope I've answered that question. So can we please kindly hold on to our question and I just round up with the last part of this post. Thank you.
don't worry. I will provide the contact of our facilitator to. If you have more questions, you can reach out to her directly.
Okay, so
Now you can conclude, ma'am.
yes, sir. Just trying to go to my slide. Okay, so this is the last part of this course, and which is more than just the GIT. That is, we're trying to look into other part of the human body that we can actually get a microbiome from. Apart, we've been talking about gastrointestinal tracts for long. So here in this part, we'll be focusing on the microbiomes outside the guts. And also, we will try to elaborate more about how a single body parts have a distinct microbial composition. So, okay. So, a study carried out by Mosimo and Mark has shown that periodontal disease that affects soft tissue around the teeth and caries, that is the unique feature of the dental arch tissue, may result from the invasion of relatively minor microbial communities. So we're talking about oral or microbiome here. So this is this major tongue, and we can see the different part of it. The tongue has its own microbiomes, the lips has its own microbiome, the inner cavity of the mouth has its own, and beneath these lips, the tissue, the soft tissue there has its own microbiomes, and all of this plays a vital role because they have the roles they play. So right here we have the caries, and you can see that this particular side is kind of invaded. Like we can see these dots that looks like disease. So we have the periodontitis, which causes it is a disease, right? In which there is especially when you have swollen gum, and as time goes on, if care is not taken, you didn't take any medication or anything, it start giving you these periodontitis pockets, like it's a hole. So and we have this, and so right here we have the oral cancer, that is the one on your lip. I think even me as a person, recently I had a canker sore on my lips, and it was there for about 2 weeks. I I couldn't get anything to use to make it go away, and it got so swollen and very red and all that on my lips. So that is a case study. So it also causes mucosal disease. That is, let's assume you're kind of using some mouthwash or maybe some kind of toothpaste and you're reacting to it or something. So it could lead to all of it because it kills the natural microbiota in your oral cavity that helps to make it healthy for you, that helps to fight against whatever, like that stops you from reacting to whatever you eat. So once these niggers are dead, they're not there anymore. What happened to you? You're so susceptible to whatever would happen. So right here, we have, okay, I said not only does oral microbiome differs in health and disease state, it is also different in composition from one intraoral site to another. So this diagram we have illustrate better the different parts of the oral cavity, and each part has a distinct what's it called, microbiome. Like this is the tooth surface, so we can see that for the tooth surface, these are the niggers, the chalice that are actually that colonize the side, and we have the gingiva crevice, which is here. You see, we have this babies, the fusobacterium, that is where they stay, and we have our oropharyngeal, this inner the upper part of your jaw inside. So we see we have all these what's it called? We have all these mummies and daddies and grannies. That is where they stay. That is the natural what's it called? community. So we have this when you have a dental plaque, for instance, maybe you kind of have a toothache and you realize that so for some people the part of the teeth will be kind of cakey. So we have these are the likely organisms that that you can find that these niggers right here. And when it comes to your tongue, we have all these. So this diagram just shows us that different parts of our oral cavity has different microbial community. Different part of it. You can assume that it's not just my mouth, but each part of your mouth has a different and distinct microbial or community. So the tongue has been identified as the most popular niche and has a significant impact on other agents in the oral cavity because it is this tongue you use to lick whatever you want to lick, even when you want to swallow your food. It is this tongue that you use for everything, so it has impact, more impact. That is why medically they'll tell you that it's good to always wash your tongue, to always wash your tongue. So okay, so this right here, the oral microbiomes is kind of associated to some gastrointestinal diseases. So that is why they say that it plays a vital role. So we can see that it causes this rheumatoid arthritis, diabetic, it causes cancer, atherosclerosis, Alzheimer's diseases, because obesity, HIV, liver, like because so many, even the inflammatory bowel disease, especially this one, I think most of us will be familiar with this, where you kind of eat a contaminated food or maybe something that you're reacting to. Either you start stooling, like you have excessive diarrhea, maybe let's assume you ate a food that is being contaminated with salmonella, and this organism causes salmonellosis, then you start purging excessively and all of that. So got an inflammatory bowel diseases. And let's assume, naturally, me as a person, I don't like eating salad out, and the reason is that the salad, the leaf, that serve as the natural community for Clostridium botulinum that causes botulism. And you realize that when you eat a food that this organism is contaminated with, what this organism does is that it causes neurological, diseases that if care is not taken for immunocompromised patient, it will lead to paralysis and afterward it leads to death, right? So it's most of the things it causes. And right here you can see it causes polycystic ovarian syndrome and adverse pregnancy outcome. For some people, it leads to miscarriage for them. So that is why whatever we take, whatever we really, really need to take care of it. Like this time around, it's not about just science bullshit. It's about when we look at science, you realize that it relates to everything. Science is you. Science is everything you do. So right here, we can see that this and the microbiomes community, the community of microorganisms in this and is totally different from what you have in the food, like totally different because it's different body parts and each this color code you see here shows you the different, it signifies different part of the body and the different organs, and you can find that. So you can totally see that for the one of the food, it's what you see here is not the same with what you see for the and we have more bacteroidetes, and for the food we have let's see the color code. We have the other Firmicutes and Staphylococcus, like we have these niggers right here, so it's different. So this slide right here is trying to focusing on respiratory microbiome. So we've talked about oral microbiome. We've talked about one of your foods. So we're talking about the respiratory microbiomes. So in 2013, majority of deaths in children of age 2 to 6 years old were resulting from infectious disease, especially pneumonia. And this graph we have right here shows us that like it was really, really high, and it leads to so many deaths of children. And right here, the conventional mechanism of the pathogenesis of bacteria and pneumonia is that the lung is sterile and there's a large inoculum of bacteria. So right here, so let's just ignore that this whole thing is cleared out. The lung is meant to be sterile. But because there is a large, what's it called, inoculum of pathogenic species, you realize that this it enters from the sea. That is why whatever you take is very, very important. See, it enters through the outer cavity, right? And it goes down and down, down to the lung, and from the lung it transports itself to the upper trachea, and it goes and goes, and it gets to the lung and causes whatever it want to cause. So it causes when it once get it causes a pneumonia that is the alveoli of the and the surrounding lungs get infected, and this leads to pneumonia. And this has been what is it called? The leads to deaths of what's it called? Children of 2 to 6 years old in the year 2013, and this statistic like the mortality rate was crazy, like about 44%, which is really, really crazy. So right here we see the that is what Dr. Asher was trying to say the other time when he said that that is why those, especially those babies that are being given birth through C-section, they advise the mother to breastfeed them properly. So let's now forget about whether vaginal born or C-section. Naturally, you realize that for the breastfeeding mother, the doctors, the health practitioner makes it compulsory to tell them that it is important to breastfeed your baby very well, because by the time you breastfeed your baby, most of the microbiome from the mother womb automatically goes to the baby and help the baby. That is why it is really, really, really good. So right here, see, we have the breastfeeding practice. This is the more when you breastfeed it goes in the milk. So look at what it does: fatty acid, hormones, and so many things like that. So it's really, really good, because right here we have the milk microbiota that are there. So the moment the mother is breastfeeding the baby is getting it. The baby is getting it. And right here we can see it. Like this looks like a kind of fallopian tube. See, as this is going in, and that is why I tried saying the other time there, for ladies, we know that we have some natural, some organism that the vagina is like the natural habitat, right? But by the time you start using soap, all this solvent, so many things to keep washing and rinsing, you tend to get rid of them. And when that happens, we're prone to toilet infection and all of it. And before you know it, you start having some itching, foul smell, and all of that, which doesn't make sense. So, okay, that is what I'm saying. This side is, but this is it's a starting result that shows the bacterial vaginosis plus. So this is when it's absent. So we can see that in this population, like it's really, really high. Like we have this diverse microbial profile. This side, why this side is dominated by two lactobacillus because and species. So apart from that, we can see this plots we have here shows us for the lady that was asking that why do we have to study this? Why do we have to know this? And you realize that in 1980, my mom research, nobody was really interested in this. They don't, maybe they don't even know much. And you see, as time goes on, gradually, we realize that the initial peak took up in the year 2000. That is when people started getting the gist. Oh, I think we need to study this. We need to know more about this. When the diseases was, getting really, really high, and you can see as time goes on, it keeps going and going. And can you see the massive peak in the year between year 2015 to year 2020? See the various, when you check your PubMed to search for the time 16 sequence, and you receive so many research, so many studies that has been conducted. This prove the importance of this testing as RNA, sequencing technology, because it's a diversified what's it called, techniques that can be used in different facet of science, whether you're in the medical or you're in the environmental or you're in the agricultural or you're in the what's it called, maybe the food industries, and wherever you are, it's really diverse. So it's a good technique. So, and which means that as time goes on, when people keep making more mistakes, they'll be available um, hypotheses, there will be new discoveries and all of that. So this is our negotiation. So like I said the other time, the 16S RNA is from bacteria. And the conclusion, microbial community exists outside the human GIT. The slides are proving that, and also microbial community are different for different body size, and we even find different communities within different topography of the same body. The term microbial diversity is defined as the number of different organisms and how evenly they are distributed within an environment. And I think that was what Dr. Ashram was also trying to say the other time. So in numbers of studies, in numbers of studies have shown the microbial communities detected on different body cells are associated with health and disease states. The term microbiome is an umbrella term for a number of microscopic organisms, which include bacteria detected in any given environment. And last but not the least, a 16-base RNA isotope sequencing is used to study the bacteria profile in any given environment. And so this actually comes to the end of this introduction to 16S RNA microbiome analysis. Thank you for listening.
Well, thank you so much for um, the in-depth, insightful, knowledge-packed, and exciting delivery on the 16S microbial RNA analysis introduction. I know there will still be some questions, but I would want us to write that down, and then if time permits, after our second presentation, and I'm very sure some of our questions would have answers even in the next presentation because of the relationship both has with each other. So it will be, I would want to use this medium to first of all appreciate the presenter for taking out our time out of our tight schedule and also prepared and researching. Although it's not a big deal for her. That's what she has been doing. So, but notwithstanding, it takes a lot of work and effort to put together this great delivery. Thank you so much. I really appreciate you. And Mr. Akin Meiji is raising up his hand, please, so that we can give the next presenter the time. I'm very sure that most of our questions would also be answered by through the next presentation. So thank you so much. I really appreciate. So, Dr. Abdul Qadir, are you
Sorry,
there?
I take a
Yes,
bow.
I'm
I'm
here.
really closing out already. I've been up all through the night. Please.
Oh, sorry.
Thank you, sir. Please, can I take a bow, please?
Okay, no problem.
Thank you. Thank you, sir.
I will share your contacts with the group. So if more consultation and inquiries, they will reach out to you.
Yes, sir. Thank you. And thank you, everyone.
You're welcome. Dr. Abdulkader.
Yes, sir. Can you hear me?
Yes, loud and clear, sir.
Okay.
You have the floor. We want
All
to have
right.
every. We want to have you 100%.
Okay. Thank you.
You're welcome, sir.
I'm trying to share my screen.
Okay. Mhm. For those just for those that are just joining us, this is the next session, and it's will be a little bit of hands-on, and we want us to prepare and take every bit of the training.
I don't know. Can you see my screen?
Yes, yes, yes.
All right. Uh, Thank you, listeners. I know it, you must be tired. Uh, you have been listening for more than 1 hour. So, in order to compensate that, since there are no drinks to refresh your minds, I will definitely try my best to shorten the time and to make it more precise. Can you please hear me very well?
Yes, we can. Yes, we can.
Okay. So, do you listen to a background sound? Because my CPU is giving some noise, so I don't know if that distracts your listener.
It's not distracting us.
Okay. So as my senior colleague present about the microbiome analysis, so when we look at bioinformatics in general, it's kind of diverse area of research, which include part of biology at large, and mostly related to diseases and how we can understand the pathogenesis of those diseases, and especially how we can easily give solutions uh, to, to, to the current problems without using a wet lab. When I say wet lab, obviously we know things that has to do with chemicals and some other related laboratory equipments. So various tools are being used in order to further understand the concept of a disease. One of them was the Motor software for microbiome analysis. I don't know if my colleagues have already give example of those softwares. And there are many, many other softwares at our disposal, depending on the hypothesis we want to provide. So Cytoscape is one of the softwares that we use also in microbiome analysis. Uh, but in order to explain more about Cytoscape, uh, we don't restrict the function of this software only in microbiome analysis. Rather, we expand it into gene regulation analysis, gene expression analysis, and anything that has to do with network. To explain more, Cytoscape is not only used in the life science, like biology and all that. It can also be used in social science. Uh, I believe some of you might have insight about that, but let's not deviate from the point. Let's concentrate on the main biology part of the bioinformatics. So uh, I was already introduced by my senior colleague, Dr. Ashraf, and I'm also a PhD candidate here in South Korea, studying molecular biotechnology. So these are the tables of the content from the introduction up to the last part. One thing I supposed to share with you guys before the session started, or even before the day, but nevertheless, it's not that important. You can do it later. What I'm trying to say is, I wish you guys have the Cytoscape installed in your computer so that we can do the work together. But I think we have a short period of time, and I believe the Noble Kids one day will have a complete session by which we can have a hands-on the Cytoscape deeply. Yeah. So I will just give some brief introduction, not more than 10 slides. Then we can take a look at the software, how it functions, and let's see how we can play with those genes and see what kind of result we might get. So introducing Cytoscape is just like open source software. Open source, you don't need to pay money to buy. You just have to follow the link as I show here, download it and install it. That's quite easy. So, which we use mostly for visualization of molecular interaction, also like network between genes or any other network which is outside gene interaction. Also, it helps to know the biological pathway of our gene combination, and also to integrate this network or annotation for gene expression profiling and other data. So we also use it for like data integration and visualization of other data which we can link Cytoscape with. For example, these days there are many softwares or packages or plugins which we can link two softwares together. So Cytoscape also enable that kind of things, which you can link, for example, R programming language with the Cytoscape. That means when you perform your analysis in R software, instead of you to get the result from the R, then come to Cytoscape, upload the files, and start the analysis. There is a way by which from R, you can directly link your result to Cytoscape, and you can control everything from the R software. All you need is to have your output in the Cytoscape. This is quite interesting, and I hope we can have time to see how it works. So it has various usage, including visualization, even at social network. Also, you can gather social interactions from web by a variety of web services. It means you can link the Cytoscape with other irrelevant softwares just to get visualization in the Cytoscape. I hope you understand what I'm trying to say. Let me repeat myself. What I'm trying to say is you can perform analysis with any other software, and for you to have a very good visualization, because some softwares, they don't have visualization facilities. Therefore, they just give you raw data, as in text file. But Cytoscape can help, and it helps a lot to get your visualization from another software on the Cytoscape. So also, Cytoscape perform predicting roles of group of genes. For example, you have gene expression data, and your data only show you, for example, you send your data to a company and they just give you the raw information. These are upregulated, these are downregulated, these are blah, blah, these are blah, blah. So you just see the raw data, but you don't know why this group of genes are upregulated, why this group of genes are downregulated. So Cytoscape can help you to identify the function of those group of genes. Also can help you to make a prediction. When this gene A and gene B combine together, they can serve a particular function. Cytoscape can be able to tell you this. Or when you have gene A and gene B, what is a possibility of gene A and gene B to combine with another gene C. So it will tell you what is the name of that gene C, and when they combine, how will they function. So we'll go through this, and I hope I will try to make it very easier for the beginners. And for those who are already advanced, I know definitely they'll find it very easy to understand. So looking at this, these are just few examples of what Cytoscape can do. And you can search for more because Cytoscape has no limitation. You can't believe what you can do with Cytoscape. The more you know the software, the more you know your hypothesis, the more you can play with it, and believe you me, you can get some amazing results. These are like examples of just few publications so that you can have an insight on how the Cytoscape work. For example, in this analysis, they use Cytoscape as one of the bioinformatics tools to analyze genes found in thyroid carcinoma. So when we look at this, the whole network, as a layman, what you can only understand is there are genes and there are different colors, and that's all. But when you know Cytoscape, you can give a lot of interpretation by looking at this picture alone. So the circles, we call them knots, and these lines are called edges. While obviously the names attached to the edges and the knots are your genes. So for example, I hope you can see my cursor very well.
Yes, yes, yes.
Yes, we
Okay,
can
thank
see
you.
it.
Okay, thank you. For example, just look at here. Okay,
Sure.
this one is a bit bigger. So can you read what was written here? It's tyrosine metabolism. So if there is a combination of this gene, ALDH1A3, and this and this, these are 5 genes. So when they combine together, their biological function or cellular function could be regulating tyrosine metabolism. I hope you get me right. Also, this one, that is regulation of lipase activity. This gene and other genes that are attached to this node via about 4 edges collectively perform a function of regulation of lipase activity. So this is some amazing things that you can do with Cytoscape. Also, for example, in this analysis, we did something like, we know COVID, everybody is trying to understand what is COVID and how it plays a role. And there are many hypotheses about like vitamin D and immune regulation, cytokine storms, and all that. So by using the Cytoscape, we were able to see the relationship between these cytokines as well as vitamin D related genes. So Cytoscape itself tells us that this group of vitamin D related genes have a friendship, a strong relationship with this group of immunes. Not only that, it tells us every connection and details about the connection. For example, this cytokine, CCL2, is connected, for example, to vitamin D receptor, VDR. So Cytoscape will tell us which type of connection is that. Is it a physical connection? Is it co-expression? Are they connected because they co-express? Are they connected based on prediction? Is it that the Cytoscape predicted the connection? Or are they connected because they are genetically interacted with each other? Or are they connected because they share a common pathway? Or sometimes genes are connected because they co-localize with each other in a particular cell, or in a particular, or within the cell, maybe in a particular region in nucleus or something like that. So Cytoscape will tell you every details about that. These are just few examples, just to give you more insight about what Cytoscape can do. All right, this is just how Cytoscape looks like. I wanted to give you like a raw picture without any figure, but then I think this will also be the same thing. But by default, when you open your Cytoscape, you will not see anything like this. This is just a screenshot of my Cytoscape because I have a lot of work, and I don't want to like remove them to show you the empty Cytoscape. But the main thing that I want to show you, as I said, Cytoscape navigation, how you can navigate the software. First of all, we have this toolbar where you have files, edit, view, select layout, app, and tools. So for now, what we need to focus on is the apps and the tools, because these are the places where you can import a plugin. A plugin, Cytoscape doesn't just work like that, because I told you it can serve hundreds of functions. So for every hypothesis, for every analysis you want to do, there is a particular plugin specifically for that analysis. And for that, you need this app to like bring in the tool into the software, just like install the tool into the software, then perform your function. Perform your analysis. Also, this is another very important part of Cytoscape, which is network. After you introduce your plugins into the Cytoscape, and after you put on your genes, which I will show you how you can do that, this is where you can navigate your file in the network. And secondly, this is style. For example, you can see this looks very organized, right? It has like unique pictures. The style looks diamond. They are well arranged and all that. The edges, that is the line that connected the nodes, are thin and not shouty. So you need this style. This is where you can make all these settings to make it very beautiful. It doesn't just become beautiful like that. You have to modify it. You have to make it look very awesome, because when you are going to send your paper for publication, the first thing the editor will look at is your figures. If the figures look matured, beautiful, then you give him first impression. But no matter how good your result is, that first impression will lead your paper to be rejected like from day one. So that's why beautifying your figures is always important. Also, Cytoscape, luckily enough, give us the user-friendly opportunity to edit our figures very well. So you need to concentrate on also style. So this is for now. And there is one more thing. I really don't want to take you far, because I don't want you to lose some information that are vital. After you upload your file, there are two ways where you can export the raw data. Because in some cases, you need the raw data for publication. For example, the the journal will ask you, we need this information, we need that information. So all your information, for example, here, the whole of this network, the information about this network connection is here as edge table. Also, the information about these nodes, their names, how they are connected, the degree by which they are connected is here in node table. So once you click here, you will see the complete summary of everything, and you can easily export it. This is where you can export the figure as well as the node tables or the edge tables. We'll come to that once we start to use the software. So assume you downloaded your software, you install everything, and you have 0 plugins. How you can do that? First thing, just look at here and click on app. On app. So when you click on app, this is like the first stage. It will show something like this. Don't be confused. Just go to your app manager. That's the second stage. After clicking your app manager, it will open like this. It will show you something like this. So just type the name of the app you want. For example, like I try to type Cluepedia, and it will like tell you the summary of the function of this app. For example, here they said it's a plugin for pathway insight using integrated experimental and in silico data. So I believe before you start searching for an app, you already know what you want to do. So after then, you can just click here. Here is install, and it will automatically install your plugin. That's as simple as that. Cytoscape is really user-friendly. It's not like our programming language where you have to install, maybe using bioconductor, using this, using that, and sometimes it will tell you, no, your version of R doesn't support this and that, and you'll be confused. So Cytoscape is really user-friendly. So I told you there are like hundreds of plugins that you can use in Cytoscape. But after like I read a lot of papers, and I can tell you that mostly these are the commonly used applications or plugins in Cytoscape. The first one is a string. So string is a plugin in Cytoscape 1. Also, it's a name of a database where you can observe gene interaction. So when you Google, just write string, you will see the directives to the main database for gene interaction network. We'll do that. So also in the Cytoscape, there is a plugin called string. So we'll also look into that. So what does this plugin do? It helps you to see the gene interaction. For example, you have your gene expression data, or you have like collection of hundreds or thousands of genes, and you don't know where to start. Which gene is connected to who, and what's their function? Are they related to any disease or not? Then just quickly go to string database. We'll do that, and it will show you the gene interaction network complex, and from there you can send your result to Cytoscape. I will show you how to do that. Also, once you get your network in the Cytoscape, you can use it to do a lot of things, like manipulate the genes, which gene is more important, which is like a hub gene, that is the most connected gene, and which kind of disease is related to this group of genes. It can tell you all that. That is the function of string. Secondly, there is gene mania. So this gene mania as well is a name of database as well as a plugin in Cytoscape. So the function of this, as I mentioned in my introduction, it can tell you gene prediction. For example, you have 10 genes that were told are associated with cancer. All right. So these 10 genes, you also want to know what other genes that might possibly be associated with cancer. It's a prediction. So you can use gene mania, put your 10 genes that are reported to be associated with cancer, and then predict which other genes can be associated with this first 10 set of genes. That's the function of gene mania. Also, gene mania will tell you the information, the details information about the gene interaction. Why gene A is connected with gene B, and which type of connection is that. So when you have a gene mania plugin in your Cytoscape, you can be able to play around with this kind of information, and you can drive something very interesting. The third one is Cytohoba. I don't know if I pronounce it very well. Please correct me. This one, you know, when you have condensed group of genes, you need to know like which one is most connected, because you may have like 20 genes or 20 nodes in the language of Cytoscape, and at the same time you can have 100 edges. It means one gene can be connected with like all the 20 genes at the same time, and another gene can be connected with only 5 genes out of the 20. So this CytoHop will tell you which gene is the most connected, and based on that, the most connected gene is mostly one of the most important genes. So this is one of the function of CytoHop. So there is M-code. So this plugin, it helps you to understand the models. You can generate models. When we say models, these are a group of genes that are grouped together in a bunch of like complex network because of their similarities. So M-code will help you identify those genes that are similar in function or similar in whatever the activity, and it will group them together. So by the time you have your model, for example, module 1, module 2, module 3, it means these genes that are combined to form this model, they have some peculiar uniqueness, and it's up to you. You have to go deep and find out why they are grouped together, why they are like friends to one another. It means they have something that they shared in common. So this M-code gives you this kind of information. So the penultimate one is CLUGO. So this CLUGO will tell you, for example, after you do all the analysis and you have your genes or group of genes that you think are very important. For example, the models. Let's say you run the analysis and you have 5 or 6 models, and you think these models are very important. So you need to check their pathway. You need to check their biological function. You need to check their molecular function. You need to check their cellular function. So you need a CLUGO. This CLUGO will help you check this pathway, biological function, or molecular function. And it will tell you, okay, this group of genes or models that are grouped together, they are grouped together because they share a particular pathway, or they are grouped together because they share this kind of biological function or this kind of cellular function, blah, 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 like that. So that's the use of CLIGO. So the last one, but not the least, I told you these are just the most commonly used plugins based on my area of research. Of course, I should make this clear. So this RCY3, it's used for co-expression analysis, and it's one of the plugins that I told you. You can use it from R programming language and have your output in Cytoscape. You can use this plugin from R programming language. You just have to install the package in R, do the analysis in R, and then get your output in Cytoscape. So what's the function of this one? We have these days co-expression analysis. For example, at times we hear that gene A is always upregulated in this kind of pathway, in this kind of disease, in this kind of cell, in this kind of tissue. Gene X is always upregulated. Dr. Ashraf, you want to say something? Okay, so we also want to know which type of gene co-express with this gene of interest. I mean, if gene A is always like, gene X is always upregulated, always expressed in certain disease, so we need to know what are other genes that can be friend to this gene X. Because mostly when we look at the whole signaling pathway, when gene X upregulated, gene Y may also upregulate with gene X, or gene Z will downregulate whenever gene X upregulates. This always happens in cell signaling and in signaling pathway, we know that. So this software, I mean this plugin, will help us to know which other genes co-express with our target gene. So by doing that, we need this RCY3, and we can use it from R software, because usually we analyze co-expression analysis from R software. So we immediately continue with this stage and get our output in Cytoscape. I hope I'm making sense.
Yes.
Okay. Thank you. So since I believe most of you don't have the Cytoscape right now on their system, I'm just giving the sites where you can go and download. Also, this is for string database, as I mentioned. And as I said, for R software, we need, for RCY3, we need R software. So let's see what we can do to explain more about these commonly used plugins. So are we ready to practice?
Yes,
Yes.
doctor.
Yes.
Okay. Okay, thank you.
Yes, we are. Okay.
Okay. So I hope you can see my Cytoscape. So it's a bit dirty, but don't mind that. Just look at my cursor so that you cannot get lost. As I said, how we can install our plugins. Go to App Manager, and write any plugin you want. For example,
Sorry, sir. Please, can you start again? Sorry.
Okay, okay. I'm sorry, I think I'm too fast. First, go to apps. Then app manager is the first thing you can see. Then click on app manager. Then you can see search. Type any plugin you want. For example, this is Cytoscape. It's already installed, so we don't want to do it again. So let's say I write this Animo. I don't even know this plugin, but yeah. So once you click here, it will tell you what is this plugin. So this Animo, the meaning of the abbreviation is analysis of network with interactive modeling. Lets you model biological network and compare their dynamics with experimental data. So this is like the use of this plugin. So you just have to click install as I click here. So now Cytoscape is installing the plugin here. So I think we don't need it. Maybe we can cancel. So it needs to configure, and I think it will take time. So we don't need it anyway. Okay, so any plugin you want, including Genmania, String, and whatsoever, you just have to do these 3 or 4 stages. Let me repeat it again. App manager, write your name of plugin. For example, String, I already have it, already installed, as you can see. So if you didn't install, just click here and you can install it within a short period of time. Also, I want to make this clear. Some plugins, they require license. For example, Clugo. After you install, they will tell you you need license, and they will give you a website where you can follow, put your email address, and they will send you the license through your email address. So don't be scared about that. That is nothing like scam or anything. It's always free. So in case if you want to install any plugin and it tells you something like that, yeah, just go ahead, and there is no any problem. So now that I install all the plugins that we need for the practice, let's see which one we can start with. For example, these are already like, let me just take, okay, let me take genes from outside so that you cannot be confused. I have a lot of genes here. So let me just take like this 20 of these genes, and let's see what we can have. So by the time you have many plugins, I told you focus on network. As you can see here, network. So if you have only one plugin, so it will appear here. But by the time you have many plugins, so as you can see, they will just come here. Can you see it? So let's start with Genmania. I just copy 20 genes, and here I selected Genmania. I want to work on Genmania plugin. Go to network, click on network, come here and select the plugin you want to use. I want to use GeneMania. That's the first step. Don't forget to select the organism, because we are doing various types of research. For example, I always work on mouse and humans. Most of my data came from knockout mouse or from human. So definitely I go with Homo sapiens. Even in the mouse, we have to choose which species. Is it M. musculus or R. navicus? Otherwise, you can spend the whole day doing your analysis, get a result which at the end will be like you have to put it in a trash because you didn't select the right species. So you have to be very careful with that. So for now, let's use this rat. Open here and paste. You can paste by doing Control V, just normal pasting, as we usually do in MS Word. Okay? Then do like this. Check here. Let me repeat myself. After you paste, then click here for more options. Check here, make sure everything is intact. For example, this is what you want to see in your result. You want to see predicted genes. You want to see their physical interaction. You want to see the pathway, their pathway interaction and all that. Okay? So we are done with that. Then just click search. So GeneMania now is trying to see out of your 20 genes, how many other genes can be related with these 20 genes. And in the output, it will show us the original genes that we input, and it will show us the predicted genes. So now it's working. Let's give it some few minutes.
Can you write us the step for this point?
Yes, I think I will need to put it in writing. Maybe after the presentation, I will coordinate with Dr. Ashraf, and I will give him the full details about the stages.
Okay, Sunday.
Yeah. So I don't. Okay, go on.
So please, how can we get the Cytoscape software itself?
Yeah, in my presentation, I put on the link where you can get the Cytoscape and download it.
Okay, sir. Thank you.
Okay.
Sorry, hello, Dr. Michael.
Okay, before it finish, I think the software is hooking. I don't know why.
I would like you to expatiate between the difference between original gene and the predicted gene. I got lost in that explanation.
The software is not responding. I don't know why. So okay, let me close it and restart. So meanwhile, someone can ask question before I.
Okay, sir. Thank you very much for this opportunity. I've been trying to download. In short, I've tried like two different types to download, but I'm using the Windows laptop. But I don't know if there's a specific one for Windows. I've been checking, I can't find.
For Windows, yeah. Also, I'm using Windows. So let me show you the website. So just cytoscape.org.
Cytoscape. Cytoscape.
Let me share it in message. Okay. Just write cytoscape.org.
Okay, sir. All right. Thank you, sir.
Okay.
Okay, my question, Doc. Thank you for the presentation.
Okay.
Thank you for the presentation, Doc.
We are not yet done. The software just hook up. Yeah, go on.
No, I just wanted to ask something.
Okay.
Can I go ahead?
Yes, sir. Please.
Okay. I wanted to ask the data you got, the genes you got. I saw is like you copied from an Excel format.
Yes.
The genes, were they raw sequence you worked on, or you got it from a database, or how?
Okay, like you just need genes as far as Cytoscape is concerned. But one important thing, you don't just give Cytoscape like genes that you make up yourself. Of course, for example, these are part of my research. These genes, as you can see, they are from hippocampus, part of brain cells. So they are more than, like, how can I say, 30,000 or something like that. So I just copy for the sake of this presentation. But you can get genes from different source. For example, mostly we get it from gene expression data. You have, for example, your samples you send to a company, and they check the gene expression and they send you in different file format. So you can also upload the file. So unfortunate, I don't know why. Okay, I'm shutting down the. Yeah, so you just need to upload meaningful genes. Also, you can upload in form of file. I hope you understand.
Okay, Doc, let me be more specific. I've done my analysis, wet lab.
Okay.
I have my sequence. I have my sequence now.
Okay.
I analyzed for 2 genes in a particular disease.
Okay.
Okay, I have the raw sequences in the ABI format, the sequence format, ABI format, file format.
Yes,
now, can I, is it the name of that gene now I will put on the Cytoscape, or I need to copy the whole sequence and place it on Cytoscape?
No. You need only the names of the genes.
Okay.
Yeah, and again, you know, there are different names given to genes. Some are ensemble, some are blah blah. So you need to use official names. It's very important.
Okay.
For example, the genes I just uploaded, they are all official names.
Is it from ensemble or NCBI?
No. These are from NCBI.
Okay.
But even if you have genes from ensemble or wherever, that is a way you can convert their names into official names.
Okay.
Yeah. Maybe I can help you while I am compiling the presentation. I can help you with the software or the database where you can convert gene names.
Okay, I really appreciate, Doc. I really appreciate.
Yes.
Okay, thank you so much.
Okay. So
I
unfortunate.
also have a question too, please.
Okay, please.
Yeah,
Go.
the question I want to ask is that though there was a time I logged out because of a call I had to receive. So, but I got the fact that when you were introducing Cytoscape, that is actually can be used to for networking. You know how genes are associated is one of the functions of a Cytoscape. So, in my mind, I was just asking that okay, if we want, for example, in gene expressions or gene regulations, you want to see how genes are connected. Now, is it possible for me, but maybe for example, now as an animal scientist, maybe I have some genes in cattle. You understand? I have some genes in cattle. That's a particular animal, and then I
Yes.
want to know how the genes interact with each other. Now, can I go to like a database like NCBI or Assemble and then look for a number of genes? Maybe for example, I just want to practice, and I need to maybe like 10 genes, from cattle, and then I download to those sequences of those genes. You understand? Now, is it
Yes.
possible for me to upload it into Cytoscape and then try to check how connected those genes are?
Yes, it's very possible. That's one of the things I want to show. Okay, let me answer your question by doing one of our examples. So this is string software. I gave example with a string. In case you don't know how to navigate string, just go to Google, just write string. Wait, just write string. Okay, so this is what you'll see: string functional protein association network. Okay, then just click on that. I'm answering your question, Sunday, with one of the
I'm with you,
explanation.
sir. I'm with you,
Yeah, I
sir.
want to make.
But string is another software apart from Cytoscape, right?
Yeah, but don't worry, they
Okay,
are
no
friends.
problem. I'm with you.
Yeah, so just navigate here. We have multiple proteins, right? So let me copy the ones I copied before. As you can see. So then you paste it here in a string. I choose multiple proteins. I want to see like their interaction. If you like, you can upload a
Sorry,
call
please.
by.
Sorry, short, please. Now, you actually, there is multiple protein by names or identifier, but you actually put in gene. Is it because of the expression? That's why you are choosing multiple protein?
Yes.
Okay.
Because they are already like 20, so we just want to see how they behave when they meet together.
Okay.
So alternatively, if you don't want to upload them separately, you can upload a file that has the names of those proteins here. Again.
Okay, already in your computer, right? Already in the computer.
Yes, that's already in the computer. Just navigate the direction and upload. Again, you always don't forget to choose the organism.
Okay.
So Homo sapiens or rats or whatsoever or bacteria, blah, blah. So here you go with this and select. Then you search. So immediately after you search, it will tell you put 21 query or genes. And it will tell you this is gene number 1, and it will tell you all the information about the gene. Also the second one, all the information like that, as you can see.
Okay.
Yeah. So the next thing you should do, say continue. So this is their connection.
Wow.
Out of the 21 genes, CCND2 is connected to IGF2. This one is connected to this one. So also like this. And this connection, as you can see, the edges, that is what connect the two knots together, has different color.
Okay, does it have meaning?
Yes, yes, it has meaning. But for now, we just want to export it. This is, we are doing the analysis.
Okay.
And in the analysis, if you want to go into the details, it tells you the biological process by which these 21 genes serve. They serve cellular response to stimulus.
Yeah.
It tells you the molecular function these genes serve. It tells you like they perform function as G protein coupled receptor binding. Also, they serve as signal receptor binding, like that. Also, it tells you the pathway that is affected when you have this combination of genes. One of the pathways is melanogenesis, proteoglycans in cancer,
Okay,
hyposignaling pathway.
please. Okay, those I see like a partition, like one gene, three genes, four genes. Is that the analysis of the connection of those genes, right?
Excellent. Out of these 21 genes, it's not all of them that play a role in this biological function or pathway or cellular function.
Okay.
Only, for example, here, cellular response to stimulus, only 16 genes out of 21 take part in this kind of thing.
Okay.
Yeah. So we want to export this. You click export, and immediately here, what did you see?
Send networks to Cytoscape.
Excellent. So you click here. So it's telling me that it's sending my network to Cytoscape. So immediately the application finished sending, it will show you something here. So our Cytoscape stopped working. I don't know why.
So that's why it's not exporting to the Cytoscape now, because the app is not working. The software is not working, right?
Yes, but I think let me switch to the other computer. I don't know why it stopped working. Someone should pray for the app to work.
It to work in the name of Jesus.
Amen, amen. Anyway, permit me to switch my okay, let me stop sharing. Okay. I'm trying to share from another computer. I'm sorry for the disappointment. I think it's my Cytoscape is overloaded. Dr. Ashraf, do you have to give me permission to share? I don't know. I'm trying to share my screen on another computer.
I don't need permission. You can share at will.
Okay.
Maybe it's have
Okay,
to
yeah.
take.
Come in.
Okay. Yeah.
Okay. Okay. So I believe you can see my screen now.
Clearly.
Yes, yes.
Okay. Let me. So we were trying to check gene mania. Let me see if I have some name of some genes. All right, it will take time for me to navigate some file for genes, but let me just write some names at random. People should give me name of genes, please.
ITS2.
ITS2, okay.
DRA1.
Must
DRA1.
it be in the same spreadsheet?
Okay. Oh, sorry. It's not the same species.
Don't worry,
I think
just give. It will fish out. It will fish it out.
Okay. Can add COX1, sir. COX1.
Okay.
it's not a gene, no. It's a marker. Genetic marker.
Anyway, we just want to demonstrate. So,
myostatin gene.
So we select mouse and we confirm like this is okay, and we search. So once again, this software will predict some other genes that are related to the genes we already put in.
Sorry, excuse me, please. I think maybe I did not get this part. Is this another software or the Cytoscape also?
This is Cytoscape, but I'm using another computer because the main computer, like the Cytoscape, crashed. I think.
Okay, no problem. Yeah. Okay, is it the gene mania? Is it a particular option in the Cytoscape to look for related genes?
Yes, it's a plug-in. It's a plug-in. I show you from the beginning how you can have a plug-in, right?
I did not get that, please, because I was not around, so I
All
was
right.
logged off.
That
So
don't
they have.
pass you. So yeah.
Okay, the Gmania. Okay, is the Gmania a part? Okay, at the right side of the software, right?
Yes.
Okay, I will click it and then I will put a particular name of a gene there, and then I will search for the related genes, right?
Exactly. Just search. So the dark one is telling us our input genes, while the light one is telling us our predicted genes. So this is how gene mania works. And
okay.
I told you last time about not tables. This is not tables. You can scroll like this, open. You'll see all the information, the gene names. These are the query. These are query and predicted. The name of the organism, shared name, log score, how they are connected, ensemble ID, all that. This is edge table. It tells us how they are connected and all that. So don't forget to navigate from here. And another thing I told you before, this software can tell us which type of relationship these genes have. For example, when you see this color, it means physical interaction. When you see this color, it means the genes are connected based on prediction. This color says they are co-expressed, mean when one is present, the other one will be present. When one is absent, the other one will be absent. All and etc. Genetic interaction, blah blah blah. So this is like in brief how gene mania works. So the next software we want to use is String. So as I told you, let's go back and try the String again, so that we can see how it works. So just go to Google and write String. Go here. Okay, so go here. I did all this before. So let's, oops. Okay, let's copy the name of our genes. STR. One thing you should know, the maximum number of genes String could accommodate is 2,000. When you have more than 2,000 genes, you need to adjust.
Okay.
Yes. So just do that. It's very easy. Select the organism. Don't forget with this.
But sir, the gene name will change it in database to other. Can we just use it the sequence?
Sorry?
The gene name will change between database to other, like NCBI or other. Can we use it the sequence for gene?
Yes, you can use. That's why in String you have many options actually. So here we choose this. I will show you again. Here, if you are afraid the names will change, you can use sequence. But now, since we have this, we are using multiple proteins, but you can use sequence, blah, blah. You have many options, actually. Yeah.
Okay, sorry, please. So the names of genes we copied was actually the names we used in the cytoscope gene mania, right?
No, we finished with gene mania.
Yes,
Now
I know we are done. I
yeah.
was wondering where the names of those genes were copied from.
No, I just use them as an example. We write them up here.
Okay, somewhere, and then we copied it down.
Yes. If you have your data, as one of our brothers says, he has some data, so you just have to use those data for your analysis.
Okay.
Yeah.
Thank you.
So now we are trying to search for these genes. As I told you, the string will tell us about each and every gene, as you can see here. So we don't need this, just keep on moving. Let's see the connection. Nice. So as we can see, 5 of the genes are connected, while this one, he doesn't have a relationship with them. But these 5 are connected, as we can see. So we want to view this connection. So we have to export here. Export and send to. Oh my God. So my Cytoscape is already open. The database is asking me to open, or I should install the latest Cytoscape. And we don't have to. Come. Anyway, we don't have time to upgrade the Cytoscape. That's why I use my desktop, because I am using the latest one there. So this is just to demonstrate to you, if, for example, the Cytoscape I'm using on this laptop is latest, the network will directly go to Cytoscape. But unfortunately, it's not latest, so they are denying to send Cytoscape. Anyway, this is just to show you how the string works. Alternatively, if you don't want to use that string database, you can use directly here. That is string database. I'm repeating myself. If you don't want to use it because of this kind of issues, you can use a string plugin on Cytoscape here. I already have the plugin. Let me show you. So we are going to check protein query. We paste our protein here. And here, there is one thing. After you change the organism, don't forget to change the organism. Happened to me actually. I did analysis with a human instead of my lovely rat, and I had to repeat for, like, I wasted many days. So I'm always curious about that. So if you are looking for protein interaction, please listen carefully. Your confidence score should be 0.4. If you are looking for protein interaction, your confidence score should be 0.4. But if you are looking for disease, it should be 1. Am I making sense?
Please repeat.
If you are looking for
Yes.
protein interaction, it should be 0.4. But if you are looking for disease, it should be 1. You should adjust your confidence score to 1. But now we are looking for protein interaction, therefore we leave it at 0.4. Right? So that's all. And we click search. So also the advantage of string on Cytoscape is that it can take more than 2,000 genes, while the one on the database, it can take less than 2,000 genes. So this is our result. This is our result. It has no difference with the database, right?
Yes.
Yeah.
Exactly. They're
Just,
similar.
yeah, similar.
Yeah.
Look. Okay.
So one thing, you should come here, string, click string here. You can change the looking by adjusting here. As you can see, I'm changing the looking.
Okay.
So the gene that doesn't have a relationship with them, we can take it out, as you can see here. Did you see?
How did you remove it?
No, please repeat, Doc.
Look at my cursor. Just come here, you will see string, click string, and you can navigate everything. You can change all these kind of things. And the gene that has no business with the network, you can remove it by clicking here. You understand?
Okay.
Yeah. So this is string. because I'm using just 5 to 6 genes.
Hello, I have a
It will
question.
stay. Okay, sir.
Yeah, the confidence score. Why are we using 0.4 for protein interaction and 1 for disease?
Because we don't want to make a mistake with a disease.
I think you are dealing with life. That's the
Yeah,
Okay, but for the protein, something is okay.
yeah. Because
Okay.
for protein, like we don't want to we don't want to push the software so that we cannot even get anything. But for disease, we want to be very specific because it's a disease, and like one protein can change the outcome. So we just want to maximize the efficiency.
Yeah, the
Okay.
confidence is very high, like
Yeah,
100%.
exactly. So this is for protein. Let's try the disease. Okay. Okay. You see, I forgot. Okay, here it has no. You don't have to choose the species or disease. Then just go for it. Yeah, your query returned no results. So it's difficult to have result for just 5 or 6.
What
You need like
disease? Okay.
yes, yeah. So I hope if you are dealing with your data, you can try this. Definitely will get something. So then, Dr. Ashraf, do you still have time?
Yeah,
No.
it depends on how deep we want to go further with the analysis. So I wouldn't want us to go so long and we will lose a lot of participants.
Okay,
So
I think the best thing, because as we can see from the caption, it's just introduction.
yeah, and since it's a depth that we cannot swing everything within few hours. So it will
Yes,
be counterproductive if we try to go further.
exactly. So I think it's important to stop here.
Mm.
At least now we already have the basic, and we know how to navigate Cytoscape, and uh, this software enough can give you a lot of papers if you have some data. So we can attend some questions and
Actually, okay. I'll be the first person to ask question.
okay, sir.
Wow.
Thank you so much for the presentation. As you are presenting, you can see I'm nodding my head like a gamma lizard. Because I want to be like Oliver Twist, like having it, but it's not possible. But it's as if I should copy what is in your brain and put it in my head. So, regarding the analysis, thank you so much for the. It was calm, it was gentle, and everything was coming after the other. And may God continue to bless you and grant you your heart's desires. So
Thank you,
my
sir.
question is, um, okay, should I ask it here or I should see you in camera? So your recent publication using COVID-19 and I think there's another gene that you worked on. I know it's
Yes.
not your recent. I know you must have published another one after that one, or some other ones are still under review. So how like how can we apply that idea into other disease or into other scenarios? Like, okay, let me put it this way: what ge- what prompted you to carry out that research, and how instrumental was Cytoscape to that research for that analysis that you carried out? I think that's That's a better way to structure the question. That's my own question, sir.
Okay, actually, when you look at the situation of COVID, there are many hypotheses, and even scientists, high-rank scientists, are really confused, like which direction shall we take towards the therapeutics. So, but generally, everyone believes that there is cytokine storm in every COVID-19 patient, at least with 90%. From several data, they say 90% of COVID patients are suffering with cytokine storms. So, looking at the basic biology, like whenever there is destruction in the immune system, the cytokines tends to be aggressive and they come up to fight the foreign body. So that's like the meaning of cytokine storms. And at the same time, there are hypotheses that says the COVID usually increase during winter, the spread because of cold and all that. So therefore, some scientists started thinking that, so what is the opposite of winter, which is summer? What makes summer differ from winter? Exposure to sunlight. In winter, we have less sunlight. So then I started thinking, okay, we have cytokine storm, and now we have sunlight, which is like a red line that differentiates how the number of cases is skyrocketing. Then I read a lot about the sunlight towards our immune, how it boosts our immune and all that. And I found out that there are 35 genes that are peculiar to vitamin D. These 35 genes, they always play a role with vitamin D. And vitamin D, for general information,
The way you just showed us, the way P53 is related to II3, like, is it that way? Like, the way they are related?
yes. Actually, it was started like a crazy idea. I told myself, I think these vitamin D related genes, 35, they may have relationship with these cytokines in human body. Because when we have sunlight, we have this energy coming from sun, and we have receptors from our skin that receive the vitamin D. So those receptors, they are called vitamin D receptors. And once our body receives those receptors, there are 35 genes that automatically become activated in our body.
Oh.
So I took those 35 genes. I said definitely they may have relationship with these cytokine storms that is happening in COVID-19 patients. So then I do the analysis and I perform one of the layout. I use one of the layout that help you. It helps you, like, you combine the vitamin D genes and the cytokine and the cytokines that are commonly found in COVID patients. So you combine the two, and you tell the Cytoscape, okay, distribute these combinations of genes based on their functional relationship. So luckily enough, Cytoscape told me this group of genes, they are functionally related. And in that group, it contains cytokines and some of the 35 vitamin D related genes.
Oh.
So when we check further for their function, we identify that they help in chemokine, they help in chemotactic activity.
Oh.
So biologically, when we say chemotactic activity, it's the way these cytokines travel, whether faster or slower, towards combating any foreign agent. That means in the presence of these vitamin D genes, the cytokines will travel faster to overcome COVID. While in the absence of these genes, the cytokines could not travel faster. That's why before they fight the COVID, people will already become sick, and they started seeking for a serious medical intervention. So
I want to ask, maybe you've done it as part of your work, or is your next line of research. Like now, there are some functional foods that assist in the production of some genes, if I'm right.
Yes.
So have you thought of ways in which, because when we talk about the therapeutic approach to combating disease, so especially in Africa, whereby the medical intervention might not be accessible to everyone.
Yes.
So I think your idea, if we can come up with a kind of local foods that are common, that are cheap, that would aid in the production of those genes that would combat disease, that will combat COVID-19. I think that's a research idea that you can pursue regarding, if you've not started already, and I would love to be part of that work.
This is a novel hypothesis, and I would like to discuss more with you.
Thank you. Thank you. I'm happy. Thank you so much.
Actually, the Ashraf always have novel ideas.
I think I like this interaction. I know everybody will also benefit from that.
Yes, I realized, like, science is just like it's like an art. You just have to think like a crazy person, apply what we think will work until they say this is a failed hypothesis. We should never give up. Yeah.
Hello, Doc.
Yeah, hello.
Hello. Thank you very much for the lovely presentation. I'm Mohammed Adeomi Abdurazak. I would actually, the Cytoscape was actually the main bone of my contention that made me actually engaged in this program that was hosted by the NOBLEKINMAT.
Okay.
Just a little bit contribution of idea on my own part.
Okay,
when Dr. Ashraf was talking about functional food in relation to vitamin D,
yes,
I was kind of going back, remembering that this early moonlight and the early sunlight,
yes,
helps in the synthesis of vitamin D,
yes,
through the skin, and looking at the metabolomics, which is the pathway for the synthesis of vitamin D, which we cannot take away sunlight from. I was now relating it down to calcium. Probably this is just an hypothesis coming up from my own head, just thinking, trying to see a linkage with the food aspect, which means somehow there's a absorption of vitamin, and these are chemotactics, which when we look at the signaling pathway, there's a role somehow calcium is being played in the movement of the chemotactics and the chemokines. So I was just trying to pull certain things together, and basically I will actually, just as Dr. Ashraf said, I would really like to get engaged in the next point of your line of research with Doctor. Also, I'm a little bit interested in it. Maybe we could find time or through your contact, then we could work certain things together. I would like really to be involved in that work.
Yes. Thank you very much. Actually, in that mechanism, calcium is one of the hallmark of vitamin D.
Yes.
Yeah, and that's why when someone is having COVID, usually experience joint weakness.
Exactly.
I read it and I put it as part of my, because there are some genes actually fully involved in the calcium metabolism related pathway,
Metabolism.
and they are also part of that network that help to promote the cytokine storms or to drag them back. So I experienced COVID myself. I was contracted with COVID.
Okay.
Me too, doctor. Me too.
Yeah, while I was laying down on my bed, I can really feel my theory.
The symptoms.
Yeah. So that's, you're 100% right. And you can coordinate with my professor, Ashraf, and we can work together.
No problem. I'm here in Nigeria Federal University, Lafayette. Thank you very much. I think we will contact later on.
Okay, nice to meet you, Mr. Muhammad. Yeah.
All right, thank you.
Thank you, Dr. Maigoro.
Well, thank you very much for this wonderful and insightful
The
presentation.
is back.
Remember when
You're
we asked him,
welcome.
thank you, sir. Thank you, sir. In short, my question, I just need a clarification because when you asked us to mention different kinds of gene, I mentioned COX-1, someone said is a marker. Please, I want you to clarify the difference between gene and marker. And if possible, cite example. I need this clarification because I'm really learning a lot on this platform. Thank you so much.
Okay, all markers are genes, but it's not all genes that are markers.
Sweet.
Okay.
Sweet answer.
All markers are genes.
Yes, but it's not all,
but not all genes
that are markers.
Okay.
Yeah.
Please expand.
Okay, for example, I can say, what should I say? All university teachers, we consider them as lecturers,
Okay.
right?
Yes.
So say all university teachers are lecturers, but it's not all the lecturers that are professors. You understand? Markers are genes that when you see them playing a role in a particular setup, they highlight something unique.
Okay. Are we saying primer? Is it likened to a primer and the old gene that the primer is trying to detect or sequence? Please clarify me at that angle because I'm getting something now.
Like what exactly do you want to know with regards to marker? Just when you see marker, you can have marker in PCR, you can have a marker in
probably. Can I say something about the marker thing?
Yeah.
Maybe like a marker, you will like maybe something physical with you that this thing is showing something. Well, some genes they are there, but you don't like feel their presence. I think that's the difference.
Yes. Like they don't give any meaning, but for marker, once you see it, okay, like P53 is a well-known breast cancer marker.
Okay.
Whenever you are talking about breast cancer, the first thing that will come to your mind is P53.
Okay.
Yeah. So I hope I answered your question.
Please, can I comment a little?
Okay, please.
Yeah, I think maybe we should bring it to the layman perspective. Understand? Actually, markers are just like a way of trying to track measurements. For instance, maybe human beings now want to compare like 5 human beings now, and then we want to compare them irrespective of their sex, maybe with their height color. This time around, all the other pictures, we will not look at them. We only compare them. We only look at them, or we want to try to track something just with the eyes being a kind of that measurement. So sorry, please. I'm trying to charge my phone when they are playing ball. I think you can go.
Yeah, The question is actually a very good question and as answered already. Um, the there is a particular reason why they use some particular genes as a marker. So, and it has been explained already, and that was why I mentioned in the case of mitochondrial genome, there are different genes in the mitochondrial genome, but not all of them are used as markers. So I mentioned the 13 in 2 classes ago. So, but CO1 is a popular marker. So because it is conserved, it is stable, so that's why they used it as a marker, and not other genes. Hardly would you see them using COX3 or COX2 as a marker. Can also be used, those genes can also be used as a marker, but when they are you are looking for a footballer, and you have Messi, you have Ronaldo, and you have a guy that call himself Ashraf. You are picking Ashraf as the want to represent you as a footballer. So you can see you are training, you are leaving what is supposed to be used and picking another thing for your work. So I think that's the simple answer to go about that. So next question. Our contribution.
So I have a question, sir.
Okay.
Go ahead, sir. Go ahead.
Thank you very much. Thank you very much for the presentation, sir. But my own question is this presentation is on that 16 sRNA. It's not on Cryto. Are you hearing me?
Yes, we can hear you.
I need more clarification on that 16 My question is, sir, is it the 16 sRNA sequence, has it already been sequenced out that we only reference, or when we're working with a biological sample, we need to do under 16S DNA, under 16S RNA extraction from our biological samples, or I need more clarification on linking them together with the 16 RNA sequences and our biological samples.
Please.
I didn't hear the question very well. I think his network was fluctuating.
I think the question was that we mentioned 16S RNA. That's how do we get the sample? How do we sequence, or how do we get the 16S RNA sequences for our analysis?
Okay, there are 2 ways you can get 16S RNA sequence. First, you can check the fecal samples. If you want to check the using fecal, or you can take saliva, or you can take skin swab, or anything that you want to check the microbiome. These are the major samples we use, mostly fecal. So you do the wet lab and send the result for sequencing. Then when they analyze, they can send you back. Mark, you when we say
sir,
how much?
The question is on the wet lab. After getting our samples,
yeah,
we're going to do extra DNA extraction, right?
Yes. So you need to target the 16S region, because there is 18, there is 4, there are other parts. So when we say 16S, because we are targeting that particular region, V4, V3, blah 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 blah. So first you need to know like which is your target before you start doing that. And alternatively, if you don't want to suffer and you don't have resources for doing that, you can download from NCBI online. Yeah, you can use some SRA,
secondary data.
Yeah, secondary data. You can use SRA toolkit, and if you know how to do some write some code using C++, it's easy to download that secondary data from NCBI database, and then you go on with your analysis.
Okay, thank you very much, sir.
Yeah, there are many protocols actually. Yeah.
There is a question here that
Okay.
they would like you to expatiate on. I think you've answered the question about can the Cytoscape be used on viral studies?
Yes. Even on social science related studies, you can use Cytoscape for that.
Wow, it's like SPSS.
Not like SPSS, because Cytoscape doesn't do statistical analysis, but SPSS do
No,
that.
I mean the application is like SPSS, whereby it is also useful for biological sciences.
Yes, exactly.
So
Also,
then that is okay.
even the microbiome analysis, there is a way I found out recently. There is a way you can use Cytoscape for that. You can check the co-express microorganisms. For example, if you have bacteroidetes, you can check which other organism follow bacteroidetes, behave like bacteroidetes. You know, bacteroidetes is one of the genus that is the most abundant in almost all the samples, especially in the gut. So you can use Cytoscape also for that, but connected with our programming language.
Okay, then there's another question that how are the results of this bug being different from last time? Like I think that was when you were trying to do the analysis. But anyways, we can move to, can you please expatiate on predictable genes?
So predictable genes, when you have, let's say, 5 genes, we know that they also associate themselves with other genes. For example, when you have a gene which is known in cancer patients, you know, definitely it doesn't work alone. It associates itself with other genes, because one gene doesn't give a biological function. You need a group of genes to tell you about the biological function. So if you don't know those genes and you really want to find out what are those genes that are associated with this particular gene, then you call it a predicted gene. Meaning it's not part of your data, or it's not part of your sequence data, but you predict that this gene is associated with the genes you already have in your data. So we call it a predicted gene.
Thank you so much, sir. There is another question on okay, if the research can be tried on humans, then as an animal scientist, how can it be replicated or tried on domesticated animals such as cattle?
Yes, you just need data from those other animals. For example, it depends on your hypothesis, what you want. I did a research related to a particular pathway, which we believe that pathway promotes brain disorder. So someone also was working on that, the same pathway, but on cattle. So it doesn't matter, like it just depends on your hypothesis and what you want to do.
Thank you. There are other questions regarding are there no markers that are not genetic or molecular markers, like biochemical markers or morphological markers?
Morphological markers. That's more of medicine, I can say. When you say morphology, something that you can see, or in another word, we can say like phenotype. Yeah. So when a doctor wants to know which kind of disease his patient have, you don't need to do experiment like we do in lab or we do with computer analysis. You need to see those biological markers in you, as in the phenotype and other things. So there are, of course, for example, if someone has uh, liver issue, one of the markers is his eye will show yellow or something like that. I think kidney or liver issue, something like that. So these are like phenotype markers that can tell you about that particular disease.
What about biochemical markers?
Yep. What about
Biochemical markers.
biochemical markers? Actually, I don't want to talk much about that, but I don't know.
Yeah, please, can I come in at this point?
Yes, yes.
Yes, sir.
I was actually the one that asked the question, so just for orderliness, that was why I dropped it in the chat box. Now, if
Okay.
you observe, I would say, when you made the statement of that all markers are genes and all genes are markers, actually put in the chat was that like a question mark, like I don't seem to agree, even though you were trying to explain, and I was trying to kind of explain my own understanding. I'm not saying I'm right, but that is the essence to which I understand this thing, and that for sometimes we can just use some morphological markers, like for example, now you made mention of a liver disease, and then it could be the eyes that is the marker. So then when I read at a particular time online, I discovered that before the arrival of the genetic markers, the molecular markers, there have been other markers that have been used in the past, like the morphological markers, like you said, associated with phenotypes, what we can see, biochemical markers, that is not DNA related, but they are more in the metabolism of the body, the metabolic pathway, that can be used to actually track or to be able to look at the expression of a particular maybe disease or something. So I was thinking, okay, that means it's not all markers that are genetic, it's not all genes that are markers. Based on that premise, you understand? That was what I actually asked it. I don't know whether I ended up more confused, or maybe you have a different opinion concerning that statement you made, that all genes are markers and all markers are genes.
Uh, I think
Let me come in, doctor. I think
okay, okay.
the reply to that is that there are two different things entirely. So the biochemical part studies the biochemical pathways. The biochemical marker studies the biochemical pathways, and not the molecular. We don't study, for example, one of the most reliable tools or sequences to use in bioinformatics is the protein sequences. So what we do when you sequence that's when you sequence the cDNA or RNA sequences. So in that aspect, we use the computer system to monitor the pathways. But in the case of the biochemical markers, they use the biochemical tools, biochemical approach to study maybe the reactions of a particular therapeutic intervention. I could remember one of my proposals for doing my master's in food mina was that I have to study the insecticidal resistance status of some mosquitoes in Mina, Niger State. And one of the ways to study that is to use biochemical markers. After using biochemical markers, I'll move on to molecular markers. I've forgotten the names of those biochemical marker of a thing to study all those differences. So they are not, you can't put them in the same category. So they are two different things entirely. Also, in the case of morphological markers, the morphology, for example, I mentioned the fact that there are what we call biotypes. Biotypes are insects or organisms that have the same structure, same features, but their genetic makeup Even their genetic makeup can be similar, but they cannot mate. So in that place, you can't use morphology to differentiate each other. Like now, there are different types of mosquitoes. For example, common ones. We have the Culex, we have the Aedes, and we have the Anopheles. The three of them have different markers to differentiate one from the other. So, they are, for example, in the Culex, you can start differentiating from the way they lay the eggs. So Culex mosquitoes, the eggs are always attached to the Culex. Their eggs are always cropped together. The same way with a little bit similar to that is that of Aedes. But Anopheles mosquito egg, their eggs are scattered. Also, the way they settle, Anopheles mosquito used to settle and their abdomen will undulate like almost 90 degrees to the point they are sucking the blood, whereby the Culex are not up to 90 degrees. So these are different markers. These are different morphological markers to differentiate between organisms or differentiate between two similar things entirely. So the three are different. So molecular markers are different from biochemical markers, and biochemical markers are different from morphological markers. I think that answers your. It's not. Don't be confused about the explanation or how it's being mopped up together.
Okay,
So,
actually, sorry, please. Actually, that is actually why I raised that question on the chat box, because for me, if it's based on that explanation, which is good in the context of molecular markers, you understand, we may be correct. But if we think that all markers are genes, then there are other markers. In fact, look at the the mosquitoes you just said. So even by seeing the ways those eggs are laid, you can actually predict that okay, this is the type of mosquitoes that is actually around here. You understand? So I think we need to know that it's not only molecular markers that we have or genetic markers, because
yeah,
for
they're
it to be a
different.
genetic marker, it has to be a gene. And if it's no gene, you understand? So there are other markers, morphological, biochemical, and some other ones that we can use. And thank you very much for the opportunity. I know Sabiu, the little I know.
Yeah,
We will
actually,
take
you are right.
one more questions and contributions so that
Okay,
we
can
can
I
score
make
it
a
again.
contribution, Doc, please?
Okay, I think you contributed before. If there are
Yes.
any new contributions,
Okay.
we would like to take that. We'll give that preference before you. So
All right.
if there are new contributions,
Okay.
and
Doc.
is there any going, gone?
Hello.
No, Mr. Muhammad.
Okay.
Someone came
Thank
up.
you. Doc. All right. All right. The person should have
Now
the
go
floor.
ahead. No, you go ahead. After
Okay.
I will make exception this time around, so the person will talk after you.
All right. Thank you, Doctor. Thank you for the wonderful presentations. Actually, it's just about the marker. I would like us let's refer back to the central dogma. That is the introductory part of it. Though generally markers are just things that could actually point us towards certain things to make a kind of an inference towards something. When we look from the central dogma coming from the let's say from the genetic, the genome, the DNA, coming to protein, then definitely the expression of it. So we shouldn't take the terminology, taking them actually. We don't need to that's Mr. Eludo. We don't need to confuse ourselves on that. Because the most often we take it that the phenotype is definitely being expressed from the genome, following our central dogma. So now depending on, definitely we know that is not all genes that are being expressed to RNA is actually going to succeed to protein level. And it's not all also that will be succeeded even to be expressed at the main morphological level that our eyes can pick. So it's just a level of what I just see is a level of variation in terms of the visibility now, in terms of each of the target. And the molecular level is more like the smallest part. You now move to a little microscopic part again. Then before you now move to that macroscopic one that we can now visibly see. And that's why we have various areas of expertise. Because definitely without the DNA, no protein. Without the protein, you can't talk of what you are now even seeing at that big, at the macroscopic level. So at each of these areas of focus, there are markers that is being picked. For example, at the anatomical level, if I should see like a boy, it's a kind of physical that the doctor is going to examine. But however, from the biochemical level, the doctor cannot just really maybe assume that this is just a boy. He has to take some biochemical tests to now test to check certain protein levels, maybe knowing that maybe a particular protein, when it's highly expressed, okay, this one is related to this particular pathology. So that's now a marker. A marker boy, now another marker, a particular protein at biochemical level. Now when you want to dig going further to the next, the first basis of the central dogma, going on DNA, molecular level. Rather than going by biochemical, or you can still add to biochemical test again, go further for the molecular studies. There you now see certain genes are being expressed. So all these will now confer a proper judgment for the doctor, in this case, to now make a kind of from an informed diagnosis. So these things are actually interrelated. So the marker of a thing is not really confused, not something confusing, but it's just like little areas of, but you should know that all these are connected from the central dogma. So that's just my own contribution on this, please. Thank you.
Thank you so much, sir, for that brilliant contribution. I really appreciate it. Thank you so much, sir. Our last contributor or question for today, before we call it a day. Oh, that's interesting. Thank you so much. I think.
Okay.
Okay, Mr. Muhyiddin.
Yes. Actually, mine, it's not much of a contribution. It's just to thank the presenter for a wonderful presentation. Actually, in the chat box, I was willing to ask for his contact, because hopefully I may start my PhD soon in molecular medicine, so I may try to start disturbing for some advices and some clues to do some tricks.
You will pay
That's why
for
I was asking for this.
consultation. You will pay for consultation.
Yeah, now will be decided later. So thank you very much for this wonderful opportunity. You are doing a very great job. May Allah bless you all.
Okay, congratulations, Muhyiddin, and I wish you the. Okay, PhD, the beginning is always hard, but I hope you will find it easy.
Inshallah. Thank you.
We hope you find it hard, but we pray that you have the strength to surmount the challenges. Because if you find it easy, you won't have something to say. Yes, I'm a PhD holder. So we'll pray for you to have the strength to surmount the challenges. Thank you so much, everyone. I really enjoyed today's session. I don't know where the energy came from. Initially, I thought I would be tired and they would have to come and drag me from the lab before when the meeting concludes. But today, lucky, I don't know where the energy came from, and today's event is one of my best highlights for this fourth cohort session, and I want to use this medium to thank the contributors, the participants, and the people that ask questions and make the class lively as it is. God will continue to bless you, and may we continue to rise in our chosen field and in our career. For those who are asking for how to get the certificate, I have dropped the account number and my phone number to send your full name and evidence of payment. And then I'll also make the announcement on the main Telegram group. So the certificates would be rolled out starting depending on how the volume of those who express their interest. So I will probably, if the response is high today, I would start dishing out preparing the certificate from tomorrow. So otherwise, maybe by the end of next week, God willing, so everyone will get their certificate sent to their email or their WhatsApp phone number. So I'm looking forward to everyone making that commitment, and we pray God will continue to bless us. Thank you so much, everyone. And the last day for the fourth cohort would be on Monday, the 8th of August 2022, and I am looking forward to meeting everyone in the machine learning class. So it promises to be as exciting as today's class was, and I hope that we all will be a beneficiary of that insightful session. So thank you so much, and see you on Monday by 10 a.m. So please don't mind the time on the flyer. I'm too lazy to change it. So I'm too lazy to change it again. So thank you so much. See you by 10 a.m. on Monday. And my regards to your family.
Yeah.
Doctor, I will message you now.
Okay, doctor. Thank you so much.
Thank you.
Yeah.
Thank you.
Thank you so much, everyone. I'm grateful.
You're welcome, sir.
Mr. Effiong, is that your LinkedIn name? Effiong?
No, sir. I'll get across to you, sir.
I'm going to
Okay.
search for him on LinkedIn soon.
And actually, I'm not really active like that. I'm just learning the ropes anyway.
It's a pleasure meeting, especially Dr. Ashraf. You really upgraded me. In short, ever since I joined this platform, my interest has really increased. Thank you so much. God
I'm
bless
Wow,
telling
you.
bro.
you.
God bless you, sir.
And you too, sir. And you too, sir. Let me stop the recording.